everyone. Good afternoon. Let's get started. We have a jam-packed agenda, so let's jump right in. My name is Eszter Hars, and I'm the mother of a 10-year-old SDS patient, and I'm also the president and CEO of the Shwachman-Diamond Syndrome Alliance. I'm gonna be hosting the meeting today. So sometimes we still get the question, what is the SDS Alliance? The SDS Alliance is a US-based 501(c)(3) nonprofit organization serving the global SDS community. Our sole focus is to improve and save the lives of people affected by SDS by focusing on research and therapy development. Basically, we exist to give SDS patients and their families more birthdays to celebrate. So join our global network of patients, families, doctors, and researchers working together to drive research and accelerate therapy development. The SDS POPS is our annual virtual patient summit. And the goal here is to provide patients, caregivers, scientists, and all other stakeholders an opportunity to connect with each other and discuss all things related to Shwachman-Diamond Syndrome research and advocacy. To the housekeeping items, we are using Zoom. Obviously, I think many of you are familiar with the platform. The controls are on the bottom of the screen. Many of them are not enabled for the audience members, but are enabled for our speakers. And we already provided guidance to the speakers. I'm going to talk a little bit about translation options in the next screen. If you have any questions or comments, please use the chat feature. If it's not visible on the bottom of the screen, click on the three dots. It should be in there. It's called chat or Q&A. Please do not offer medical advice, and please do adhere our community guidelines, which is basically just common sense, productive and courteous discussions. At the end, or actually following the meeting today, you will receive a survey to provide feedback about today's meeting. And the first 10 people to submit the survey will receive a signed copy of Dr. Fajgenbaum's book, Chasing My Cure. So on the next screen, just a summary of how to access translations. So Zoom now offers to show you the live captioning in one of over 30 languages. So to turn that on, you want to look at the bottom of your screen, look for the CC button, enable to turn the transcript on, and then you can choose a language. If you run into any difficulties, please message us via the chat. Next, I'd like to acknowledge our sponsors and partners. All our programs, including educational programs such as today's SDS POPs, are made possible through support from you, our donors, partners, and sponsors. We are very fortunate to have some very generous sponsors in our corner, and we really appreciate their partnership. If you or your company would like to sponsor our work through sponsorships or donations, please reach out. And according to our industry engagement and sponsorship policy, our partners and sponsors do not control the content of our educational activities or other programs, but they make them possible. Next, I would like to introduce our team for today. Jeff Cooper is our IT guru who is running the show today behind the scenes. And if you run into any difficulties, he may be the one who helps you. And also on our team today is Ashley Ferreira. Ashley is our project manager of community engagement and diversity, equity, and inclusion. Some of you have chatted with her already or engaged with her in the past. She will also help with our discussion later on in this session. Then we have Ashley Thompson. Ashley is our genetics project manager, and she has supported many of our genetics projects over the past year. She is a board-certified genetic counselor with a lot of experience working with SDS specifically and variant curation in the past. So we are very grateful to have her on our team as well. And with that, let's jump right into session 1, which is focused on patient stories. Next. So why patient stories? Why are patient stories important? Stories are how we understand each other in the bigger context of life and with everything we do. Next. So during the summer, we, the SDS Alliance, published an article in a peer-reviewed journal in which we really emphasized how Shwachman-Diamond Syndrome is really a model rare disease which deserves a lot of attention for drug development. And this is because SDS is a single gene disorder. The target organ, the bone marrow, is accessible, much easier to target than diseases where the whole body needs to be addressed, for example. And SDS was described first 60 years ago. There's a lot of data already out there. There are natural history studies. And most recently, we also got an ICD-10 code for the disease, which will also contribute to more data. And so just looking at the whole endeavor, what it takes to develop drugs, what we really want to do is to de-risk drug development. De-risking means it's just lowering the cost to make it possible for industry or researchers to actually develop a drug and take it from the lab all the way into patients. And to make that possible, likely, and fast, we need to develop several areas of the research infrastructure. We need model systems, such as the mouse model. We need to engage with the FDA and other regulators so that they know what success would look like in a clinical trial. We need to invest in data infrastructure so that data are not isolated in just individual groups, and that there's no gaps, that we really capture all the data that's necessary for therapy development. And we also need to invest into the patient community. Without participation of patients in research, research cannot happen. So this is absolutely critical, and we have a number of different programs to enhance and develop these areas. And I'm going to take you through some of these activities later on. Next. So we'll focus on regulatory engagement and patient input in this first session. So that's why patient stories are so important. So I'm really excited to share with you that we applied to hold a meeting next year with the goal, this kind of meeting is called externally led patient-focused drug development meeting. So what this is a meeting to provide the US Food and Drug Administration, the FDA, and product developers and clinicians and academic researchers an opportunity to hear from us the perspectives from individuals with Shwachman-Diamond syndrome on the health effects and daily impacts of Shwachman-Diamond syndrome, treatment goals, and decision factors considered when seeking out selecting a treatment. So these are very specific things that the FDA and drug developers would like to hear from us in order to evaluate whether future therapy is meaningful and effective for us, the patients. So this meeting is being conducted. It's similar to what FDA used to hold themselves, which is called PFDD, Patient-Focused Drug Development Meeting. But in this case, we are hosting it. And again, the goal is to systematically gather patients' perspective on their condition and available therapies to treat their condition. And then after we hold this meeting, we will also have the opportunity to compile a detailed voice of the patient report. And again, this is then something the FDA and drug developers can use to take into consideration right from the beginning what kind of endpoints, what kind of impact on quality of life is meaningful to us, the patients. So more details to come. So I just want you to remember this term EL-PFDD, externally led patient-focused drug development, and we are planning to host this next summer. Next. So with that, I'm very excited to introduce Joyce to you all. Joyce joined the SDS Alliance as Youth Ambassador earlier this year, and she has already volunteered for several projects. She is a 16-year-old rare disease advocate from Linden, New Jersey. She uses her voice to fight for governmental policy change and speak for the rare disease community, SDS specifically. Joyce goes to a VOTEC school for medicine and plans to pursue a medical career in addition to her advocacy journey. Her current goal is to create an awareness day for SDS. Handing the mic over to you, Joyce.
Hi, everyone. Thank you guys so much for coming today to hear my speech. So my name is Joyce Fitz. As you guys know, I'm a 16-year-old living in New Jersey, and today I will be sharing my personal journey with SDS and how it led me to fight for governmental policy change. So I was born shaking and needed to be fed a bottle right away prior to my mother emerging from recovery. This began a cycle where I needed to be fed every hour. The hospital nurses scolded my mother for overfeeding because bottle feeds are usually every 2.5 hours. My mother could tell, though, that my discomfort was telling me that I was hungry. Actually, I was starving. My mother knew something wasn't right. My eating during the subsequent months appeared desperate. I cried nonstop. After many hospital visits and doctor visits, I was put on a prescription high nutrition formula that cost my family $500 a month. I was also constantly sick with viruses and infections. At 18 months, I didn't wake up one morning. I was breathing, but unarousable, like a dead weight. At this point, my mother was accustomed to these unusual things going on with me. She called the doctor and brought me in for an office visit. The doctor saw my condition and called 911 immediately. I spent a week in the ICU. No one knew what was wrong with me, and my blood sugar for that episode had dropped to 36, with normal levels ranging from 70 to 100. I was sent home with instructions to follow up with Children's Hospital of Philadelphia. It was the gastroenterologist who suggested that my various problems might be Shwachman-Diamond Syndrome, and a genetic test proved him right. It was 18 months that I remained undiagnosed, suffering through the symptoms of this disorder. Surprisingly, this is a short amount of time compared to those of my friends. A diagnostic odyssey is what it's called, the period from symptoms beginning and getting a confirmed diagnosis. For SDS, the diagnostic odyssey can last years, and on rare occasions, even decades. A common topic among those with rare diseases as a whole is the long-lasting periods that people suffer without a confirmed diagnosis. Symptoms that last for years, but no specialist having a slight idea what could be causing it. In fact, the average length of a diagnostic odyssey for rare diseases as a whole is 5.6 years. Within that time frame, a multitude of complications can occur, such as irreversible damage and psychological suffering. Some solutions, which I will talk about more, include some other governmental policies and newborn screening. My parents torn between feelings of joy and being terrified. Yes, after 18 months of fighting for doctors to look into what is wrong with me, they finally got a diagnosis. A diagnosis, meaning that they have a name for what I've been going through, something to tell them that they were not, in fact, crazy, but also a disease, a rare disease, that is causing me harm and pain. a disease that they will have to manage for my entire life. A diagnosis was reassuring, but little did they know what that truly meant for my future. Soon after my diagnosis, we started the journey of managing the disease, finding trained specialists in the disease that understood SDS. These specialists would be in Children's Hospital of Philadelphia, creating a growing list as I grew up. Hematologist, gastroenterologist, nutritionist, endocrinologist, pediatrician. 5 specialists that I see while my friends can't remember the last time they even saw their general physician. I live in New Jersey, so we travel 1.5 hours to receive this care. The care of these specialists in the disease that have knowledge on the complexity of it. People with rare diseases often travel far to receive care because not many doctors are even aware of the disease's existence. Specifically with SDS, often ED doctors or general physicians never know what my disease was or how it affected me. We are lucky to have CHOP near us because that meant that some people have to travel by plane to receive care. With that being, it is still quite a hefty distance to travel multiple times a year to see 5 different specialists. From then on, I practically grew up in the hospitals and a medical setting. The nurses loved me, both at CHOP and my local ED. I would like to say that I was a very kind child growing up, always showing my generosity to others. I remember once when I brought in little ink stamps and went around to the nurse station giving out little hand stamps to the medical professionals. Or the time when I was stuck in the hospital on my birthday with an eye infection and the nurses created a big poster on my door with all of their names on it. The doctors and nurses started knowing me by name. It was wholesome. And often people criticize me for saying that I consider the hospital a second home, but it truly is. The people and the environment grew onto me as I grew up. I liked being in a medical setting, so much in fact that later I wanted to pursue a medical career. The thing about the thing about SDS is that it is an invisible illness. When people look at me, they see a healthy child, but they don't see what's truly happening internally. I grew up doing normal extracurriculars like every child does, gymnastics, dance, cheerleading. From a stranger's perspective, I was just another normal child. In fact, when I tell people my true story, they think I am joking, but I am not, sadly. People are often surprised to see that a normal person to them had so much going on beyond what they can see by the human eye. They don't think that someone who is thriving academically, socially, and creating an impact can be going through so much within their physical health. What they don't see is constant hospital visits, the multitude of specialists keeping me alive and well, or the yearly biopsies. They don't see the little girl crying at a blood draw or the teenager studying for an AP test while struggling with her physical health. They can't see the constant pokes and prods that doctors inflict on her, or the thoughts that run through their head when the doctors are explaining test results. They see me, the healthy, normal girl passing in the hallways. By growing up with this rare disease, I went through a battle of mentally embracing the person that I am and the experience that I have gone through, embracing my past medical trauma to show to the public the life of an individual living with a chronic disorder. It was my when I went to my first rare disease event, patient got to meet others like me with rare disorders and finally connected to people my age who experienced what I did. It inspired me to start getting into advocacy. I discovered Rare Disease Legislative Advocates, an organization that allowed people with rare diseases to speak to legislative officials about topics concerning the rare disease community, a place where I could use my voice to speak to those like me and my community, a space and time where I am defending my people and making a difference. This platform truly allowed for my voice to be found in advocacy. It taught me to take my experiences and turn them into inspiration. This year, especially, I've developed my public speaking ability to be able to share things that I would not have been able to a few years back. I have been on a few podcasts this year focusing on my journey and answering questions. Finding my voice in advocacy has truly been one of the best experiences I could ever dream of. Being able to tell things aloud instead of keeping them inside can truly aid in becoming one with your story. I have been featured on a hospital website and in many rare disease blogs, sharing my efforts to the world. Now, we are going to get into exactly what these efforts include and the bills that I have created and defended to Congress. The first of which being my recent passion project, which is to create a governmental official awareness day for SDS. I am currently partnered with a U.S. House of Representative member to aid me in this process of introducing the bill to the House of Representatives and then on to the Senate. I personally wrote the bill and created a one-pager, which is a short document about the bill featured on the right side of the screen. The second bill is the Accelerating Kids Access to Care Act, which would allow for physicians to easily get on the Medicaid programs across state lines. For example, if somebody is receiving care outside of state, like me, and is on Medicaid, it could be hard for the doctors to be able to be covered by the state Medicaid program if they are across state lines. This bill would make that process far more easy. The third bill is the Safe Step Act. Step therapy is a common method that insurance companies use when covering medication. It is often where the insurance company will not cover an expensive medication until the person has tried a cheaper option. This bill would allow for people to not have to go through that process and to have easy access to the medication or therapy that they need. The final bill is the Creating Hope Reauthorization Act, which would continue a program that provides incentives for drug companies to create drugs for rare pediatric diseases. They provide vouchers to a drug company that they can use to either accelerate the process of an FDA approval for a different drug that they are working on, or sell it to another company for a wide amount of money. This program has helped create dozens of new drugs for pediatric diseases and expires this year, which is why we are trying to extend the program. By advocating for these bills, I have met with U.S. senators and representatives in New Jersey and have visited Capitol Hill to advocate in person. 3 times a year, I talk to government officials to fight for these bills that would strengthen our community. These pictures represent the various times I've met in person with my NJ group full of rare disease patients, family members, and friends that help me for this ultimate goal. Today, I am more than my disease. I am someone who advocates for change, and I use my voice for a community of hundreds of thousands of people. I am a person full of interests and hobbies. I love seeing musicals, for example. My favorite musical being Hadestown, which I've seen 14 times. I enjoy horseback riding and singing. I attend a Votech school for medicine with the ultimate goal of becoming a pediatric hemato-oncologist. The truth is that all rare disease patients are more than just their disease. We are all real people with real hobbies, and advocating allows us to strengthen the knowledge of the public about our lives in a medical setting and who we truly are in the public. Thank you for allowing me this time to speak and listening to my perspective. If you have any questions, feel free to reach out or to ask them.
Thank you so much, Joyce. We are so happy to have you and so proud of all you have achieved. So let's switch topics to the next one, which is, you got a lot of applause from the audience. The next topic is our film project, which has yet to be named. But the basic idea is to shed light of what it's really like to live with SDS. That is something I heard a lot in the support groups and in between the lines, and as a parent, is that like SDS is kind of invisible and nobody gets what we're really dealing with. So this film is an attempt to show just that, and again with the goal to show people who can make a difference. For example, regulators and drug developers, so they can really understand what it is that we need help with and how to measure success, in addition to, of course, connecting at the human level. So for this section of today's meeting focused on the film, we will show you the trailer first. This will be the world premiere of the film. The filmmaker and I are the only ones who've seen it. So this is very exciting. After we show the trailer, I'm going to introduce the filmmaker, Levi Gershkowitz, and then we will switch to talking to some of the families who participated in the film. And I'm going to introduce them as well. So with that, let's see the trailer. What I'm really trying to do here is change the culture of the SDS community itself.
What this little girl is going through, she has changed the odds, so I'm going with those good odds.
Does he not feel well? We just thought, so cute. But is that him having SDS and us not knowing and him not feeling well.
I just want you to have a normal life. And at that point, I come to acceptance with my daughter's disease.
For him, he expresses his thoughts, his grief, and his pain through music.
I received comments such as, there's no way that you have that, only babies and children have that. You don't have that, you wouldn't make it past the age of 20.
A lot of the physicians really don't understand Shwachman-Diamond Syndrome due to how rare it is.
Nobody expects for something like that to happen, especially not to someone that you love. You see it with other people and you think it's something that's distant and far off and isn't going to happen to you until it does.
Living with a rare genetic condition, you contemplate death, and it's scary.
They had to come in and operate on her in the middle of our room. And Addie coded several times, and I did all of that alone.
He spent a lot of days just crying in pain. As a parent, that's like your worst nightmare. You just want to fix whatever your kid's going through.
Our patients are dying, and we urgently need new options to treat them. Well, we need a few seconds, like I need a few seconds to gather myself. But it is really, well, first of all, thank you for all the families who participated in the film. That's, I guess, the most important point. But it is really also my pleasure to introduce Levi Gershkowitz. He is the director of the film. Levi is a compassionate writer, photographer, and film director with a love for people and their stories they carry. His approach is storytelling, humanizes the experiences of people in life-limiting situations, and through living in the light, leads a team of equally insightful individuals. His passion as a patient advocate began as a child, volunteering for the NTSAD, which is another rare disease community. In 2012, he founded Living in the Light as a platform to highlight previously untold stories and bolster the unique wisdom and beauty of rare disease community. And I think you would agree that he has succeeded at that once more. Our next slide is to introduce the cinematographer, Brandon, but he wasn't able to join in person today. But just a huge shout out for the beautiful, beautiful footage he was able to capture. And you've been able to witness that in this short trailer already. And with that, I will hand over the mic to Levi.
Thank you, Eszter. I hope you all can hear me okay. Good afternoon. It's so nice to join and to see the trailer, to share it with you all, especially those of you who are in the film or your kids are in the film. So as Eszter just mentioned, I'm the founder of Living in the Light, which is a project. It started as a project and it quickly became a business, a career, and a true mission and calling in life, not only for me, but for the rest of our team, where the company is based in Massachusetts, where I live. But just to share a little bit about it, I think, for instance, I just wanted to start too by thanking you, Joyce, who just shared your story and your voice and advocacy and all the work that you're doing, because it's through my meeting and interacting with people, affected individuals, advocates like you, that I've really been called into this work. I myself, not known, living with any rare condition, and so it's through the personal connections and the friendships that form as I meet other advocates and family members that I really feel called to do this work as a storyteller. So in short, and I think, Eszter, you did a fabulous job introducing Living in the Light, but more than that, I think the trailer really shows what our work is, which is to make sure that people are seen and heard first and foremost as people, as individuals, not as a condition or their disease or even their symptoms. So often our approach is a demedicalized one, where rather than focusing on the metabolic condition, we really focus on the story. And a campaign that we've been running for several years now is actually called that, Story as Medicine, and looking at the medicinal qualities of being seen, of being heard, and having your story told. Which, if I had longer to speak, we could maybe go and look at a history, but I will just mention, I think it is important to mention that in the history of medicine, it's scientific, medical technology that we have access to today, much of the origins of medicine prior to the technology that we have existed through storytelling. It existed through a one-on-one dialogue that an individual would have with another individual, conveying what they're experiencing in their body. And because of all of the medical testing, which is quite necessary for getting a definitive diagnosis for something like SDS, sometimes in that journey, the individual either gets lost or deprioritized, and what they feel and their emotional experience, their somatic experience, maybe even their spiritual experience. And so our goal at Living in the Light is to address the isolation that many people and families experience who are living with rare diseases, and to make sure that that is included as qualitative information alongside with the very important still quantitative information that helps lead to new treatments and the development of therapies. So in support of the SDSA and in support of the EL-PFDD meeting that is forthcoming in 2025, Brandon Heath and myself are working pretty hard on this film. Currently, we have completed filming and kind of the content creation side, and so now we are editing. We're yet to be sure if we're going to need to do any more filming, just to make sure that we have everything that we need, but things are already coming along quite well. And what I mean by quite well is when I watch the trailer, I weep. And I imagine many of you did too, seeing it, because there is a very raw and tangible quality to the work that we do, and that is conveyed and that comes through to our audience, be the audience members of the FDA, be the audience families who have yet to have a child diagnosed but in the future will, to make sure again that there is a visual story and one that people can see and feel that helps to address both the individual and the collective needs and desires for treatment development and for more advocacy. So I'm so excited to see who else is here. I currently can't see anybody else's screen for some reason, so but I'm also happy to answer any questions, be them about the film or about Living in the Light in general. So Eszter, thank you for finding us, inviting us in, and also for including your story and the story of your family as, I think, a very important component in this film. I think the vulnerability of that, kind of paired with your leadership as an advocate and a researcher, is a very unique position. I can say from the 12 years of doing this work, I have met few people that wear as many hats as stylishly as you do, and really hold it all together. So we're really happy to be partnered in this, and thanks for inviting me here today.
Thank you. Thank you so much. Many hats is right. And vulnerability too. For me, it's always a balance, because what I want to do is build a platform for all patients to be able to make a difference in their children's lives. So trying not to put my family in the forefront, but I'm here as a parent as well. So thank you for summarizing that so succinctly. And with that, let's celebrate the other families too who were able to participate in the film or who actually chose to step up and really share their stories in a very relatable and vulnerable way. I'm deeply moved and grateful. Thank you. So I would like to introduce Ashley Ferreira, our project manager, who will lead the discussion. Only two of the families were able to join us live, Erika Thompson and Kassi Thompson. They sent me a brief introduction, but I think it's probably better if I let them introduce themselves. They also shared some photos of their family. And after that, we will welcome Amanda Jacobs, who will be representing Melvin and Addie, her family's story. And again, I will just let her introduce her story after Erika and Kassi, and then we can just have a discussion as a group, as a panel moderated by Ashley. So with that, I'll hand over the mic to either Ashley or straight to Erika. The floor is yours.
Well, I think that we should definitely go ahead and pass it over to Erika and Kassi and let them kind of share their story and start leading the way. So Erika, Kassi, the floor is yours.
Hi.
Go ahead, Kassi.
So our experience with Elijah was he was not diagnosed until he was in his early 20s, after he had an MDS diagnosis. We all just thought he was a normal, kid into adulthood, and was very surprised by the diagnosis. Erika?
Yeah, so yeah, I'm Elijah's mom, and we're sharing a story from a perspective in the documentary that is one of remembering and of loss. Elijah was a SDS warrior that has passed on, and as Kassi shared, it was a very late diagnosis. I on one hand feel we had the luxury of not worrying and having the same childhood issues that a lot, of parents do with the younger patients with SDS. But we did have a lot of idiosyncrasies that Elijah experienced as a child, but relatively a normal childhood existence without knowing his diagnosis. And but that luxury came at like an extremely steep price. We witnessed, many small clues through his childhood failure to thrive as an infant, many weeks in the hospital with low white cell count, not understanding why. Many tests for things like cystic fibrosis, to name, one. And every time the medical community just assuring us, oh, he's probably fine. That's strange. If I had maybe $1 for every time we heard, that's strange. He had a cough that couldn't be explained most of his life. That's strange. And just many, many clues, but always the medical community just saying, yeah, he seems fine. And wasn't until his 20s, as Kassi shared, he's starting a family. He's very vibrant in his young adult life, finding his career, having his first child, and his beautiful family with his daughter and his wife, choosing his career path, and boom, out of nowhere, he's just feeling ill. He falls sick with MDS. And, it's a very rare finding for somebody in their 20s. And I just wonder, had the medical community thought sooner, even though we were advocating, of course, that they would have done genetic testing if his outcome could have been different. And, why did it take 7 months of MDS and then, going into him getting AML, acute myeloid leukemia, for them to finally really dig into the genetic testing, and we find that he has that. And so, I don't like to should on myself. I should have this or should have that, but I do wonder what could have been a different outcome for Elijah and for our family, had that been, something they would have dug a little deeper into. And so being a part of the documentary, I'm just so honored. I want to say thank you to Eszter, to Levi and the team at Living in the Light, Brandon, for allowing us to share his story. He truly was a warrior. The next slide you'll see, we just said he was a warrior. He really was. And he approached every day his disease with so much bravery and so much willingness to still make us laugh or to be kind to the staff that came in. You see him there with these ridiculous Hobbit feet slippers, but they were a gift from his uncle. And it was just incredible that, he would wear those in feeling terrible, but making others laugh and smile along the way. He was really, truly, I say, he was this pint size, larger than life, gentle spirit, powerhouse, Yoda baby that came to us. And he just never lost that beautiful spirit about him, even walking through a very difficult path. And it's just a privilege, though, to be able to share his story in this documentary. And, I know my hope and desire is for other families, other parents with undiagnosed SDS, patients living with this invisible disease, but having these small clues that the medical community will begin to understand and think of this as a possible option, or at least think maybe we should jump to genetic testing sooner, or making genetic testing with the, all this great technology we have available in the world today, that we can maybe get genetic testing for newborns to be a more common occurrence. I mean, specifically for someone like Elijah, who was in the hospital with not understanding what was happening to him. And instead of him being in the hospital for a few weeks, not understanding what was going on, but for them to say, hey, let's do some genetic testing, how that could have maybe changed his outcome. That's kind of where my heart goes. And so being able to tell this story through this documentary, our hope is that it not only honors, Elijah's life and his battle and his warrior spirit, but also all the SDS warrior family spirits that are out there, and the ability for the medical community to feel, as Levi shared, that these are people. They're not just numbers and patients, but that they feel our life and our experience and what these people go through who are in the midst of this, and can, maybe think a little bit more about, the proper diagnosis sooner. And then, I just have to say thank you too to Eszter specifically. It was meeting her clandestinely in an online group that, led me to another mother of an SDS patient who took time and patience and just explained to me what we were going to be walking into, and making connections. Even she was like, hey, there's an online forum meeting happening today, or tomorrow. It was like right before, the day before, that we had connected and talked on the phone. And I was, was on this conference for like hours, just learning about SDS and the disease. And the community is really vibrant for this disease. And so I just want to say thank you to Eszter for creating a safe, beautiful place for us to do that together.
Thank you so much, Erika and Kassi. We really appreciate you sharing your story. With that, I'd like to hand it over now to Amanda. Amanda, if you can share a little bit about your story and your connection.
I am Amanda Jacobs, and this is Addie, and my husband, Melvin, as well. My daughter Addison is 9 years old, and she was genetically confirmed with Shwachman-Diamond Syndrome at the age of 9 months old. She was born prematurely by about a month old, and we knew right away that there was something wrong. just by her short stature. Within her first year of life, she was failure to thrive, having a lot of eating issues, had a lot of fast breathing. We didn't notice the small chest that she had until we kind of got further evaluated post her diagnosis at 9 months. And the way that we actually got diagnosed with Shwachman-Diamond Syndrome was she, at birth, she was a genetic doctor came to see us, and he was thinking achondroplasia in the beginning because of her short stature. And then going to some follow-up appointments with him, he noticed the bell-shaped chest. And he actually misdiagnosed that as another syndrome. But we went through the genetic testing. He had thought that was June syndrome. And June syndrome is a genetic syndrome that solely just has the small chest. So when that genetic testing came back that, it wasn't clinically June syndrome, we actually went through a support group on Facebook that I connected with some other parents. Addison was still having a lot of the other issues, the failure to thrive, specifically with the feeding issues. She was having some blood sugar issues. She was tube fed. And another mother had said, hey, this really sounds like somebody else that, had come into this group and I had given some advice for, and it ended up being Shwachman-Diamond Syndrome. And the mother that I spoke with, she was a nurse. So she was very, very medically well-versed, and connected me with another Shwachman-Diamond family. I went back to the geneticist and had said, hey, can we test for this? I really, looking it up and speaking with some others, I really think that this might be something that that might be going on here. And we ended up again at 9 months old, she ended up having that testing and coming back as confirmed with Shwachman-Diamond Syndrome. And with just the syndrome diagnosis, it was such a double effect there, with just the relief of finally knowing what was going on. But on the other hand, she really had a lot of the more severe side effects of it. And we really kind of knew in our heart that it was going to be, a long road that she had. So from there, we went to, unfortunately, so that all happened with our home hospital, which was Hershey Medical Center. And the advice that was given to us from our home hospital was, it's fine, she'll grow out of it. And we just knew that that was not the right answer. So what we started with was, we went for a second opinion on her chest wall deformity, because her failure to thrive was coupled with the pancreatic issues that she was going through, as well as with having such a small chest. She was breathing so quickly that she was burning off the tiny amount of calories that she was absorbing, because we hadn't figured out, that pancreas, element with it from the beginning. So we went to CHOP and got a second opinion, and we met with Dr. Campbell. And Dr. Campbell was an orthopedic surgeon who invented the vector rods, which are titanium rods that are slinged to your chest wall to with children with it. They just classify it as thoracic insufficiency, no matter what underlying syndrome or disease that you have. They put these kids mostly in this thoracic insufficiency diagnosis, and treated as such. So we decided to, at that point, she was only given a 30% chance of her chest wall growing. And if we didn't do any intervention, that they thought that she would maybe live to about the age she is currently. So with being faced those odds, we made the decision to go ahead with the rib expansions. But committing to those rib expansions, we started that journey at 1.5 years. She went into that, the first rib expansion surgery on room air with no respiratory support, a cognitively delayed child, but not to the point of what had happened after. So we went into her first vector expansion, with a almost normal operating child, and ended up enduring a 5-month hospital stay where she ended up getting both rods inserted, which they typically do 6 months apart. Both rods inserted and coming home on a trach event, having both of those surgeries. They also found that she had malrotation. She had a major bowel surgery, some other underlying issues as well. So it was just surgery after surgery, and she came out of that hospital stay a wildly different child. And so in the essence of, before we went into that hospital stay, she was talking. And to this day, she does not speak. So beyond that, we endured those rib expansions for 6 months. Every 6 months, they would go in and expand the rods, and then you would get an expansion out of those rods for about 2 years, and then we would have to go in and do a full swap out of the rods. So she went through that from 1.5 years until April of last year. They finally removed her last rod. Along with that, she was on a ventilator for 5 years, along with a trach. So going through all of those medical things and enduring all of that. I mean, Addison now is 9 years old, and she has spent almost half of her life in the hospital. But after she ended up being able to successfully get the rib expansion rods removed, get off the ventilator, get the trach out, she had been followed very closely by hematology oncology there at CHOP. And we had known since she was a baby that it was very highly likely that she would need a bone marrow transplant, because they had classified her in the aplastic anemia category, where all of her cell lines were down. She was transfusion dependent on everything, and was on Neupogen shots for her neutrophils and all of that. But because of the ventilator and because of the vector rods and all of that, she would have not survived a bone marrow transplant. So once we got through all of those hurdles, it just coincidentally, she developed a very aggressive P53 mutation that is extremely likely to progress into MDMS and AML. So last summer, we were faced with the decision to either transplant or do the wait and see game and see if it would progress further. And again, with the statistics, not in our favor with waiting, we decided to go ahead and transplant. And the opportunity that we were presented with her transplant was that there was a clinical study with a chemotherapy drug called treosulfan. And typically the chemotherapy drug that's used for Shwachman-Diamond transplants is busulfan. And with the busulfan chemotherapy, Addison only had, they gave us less than 10% chance of survival because of the side effects of that chemo. So with the clinical trial of the treosulfan, it took away almost all of the side effects, the fatal side effects that Addison would have had to gone through. And it worked. It worked very well. We actually just got the results back this past week that she had her 1 year biopsy results, and it came back that P53 mutation that we planted for is gone. And so I had put with us on my speaker thing for Eszter in my little blurb that I sent to her that honestly, I feel like right now she's healthy enough that she can start living her life for the first time. So that's kind of where we're at. We Addison still has a lot of medical issues. She sees between 10 and 12 specialties overall, and requires in-home nursing so that my husband and I can work. So that's kind of what our dynamic is. She gets day shift nursing that's up to 7 days a week, and night shift nursing as well, up to 7 days a week. So that we can, because she has medication, she's on a pulse ox overnight, her feedings, all of it. It's, all throughout the day. So that nursing enables my husband and I to be able to work and do what we have to do. And that's pretty much our story and kind of where we're at. But, kind of to echo Elijah's mom, we are very grateful for Eszter and to be a part of this documentary. It really honestly means a tremendous amount to us. We have been involved in the Shwachman-Diamond community as much as, we really can be. And we really do enjoy the Shwachman-Diamond community as a whole. They have been a tremendous support to us. Any questions that I have, and just the parents alone, within the multiple support groups that there are available, there's nothing like talking to another parent that has already gone through it and can give you that specific feedback. It really is an amazing group to be involved with. And in the film crew as well, Levi and Brandon, we loved, doing all of that and participating in all of that and, sharing our story.
Thank you so much, Amanda, and Melvin as well, for being willing to share your stories. Likewise, Kassi and Erika. And actually, I would love to invite all three of you back so that we can just have a brief discussion. Again, thank you for your bravery in sharing your stories now and in this film project. We know how difficult it can be reliving a lot of these journeys, right? And so part of the reason that we were inspired to work on this project was really to shed light on the invisible aspects of SDS so that therapy developers and regulators such as the FDA, they can gain a more in-depth understanding of the unmet needs of our community, what really matters the most to the SDS community, and the priorities that we want them to focus on. And so with that in mind, what was a specific aspect of living with SDS that you really tried to highlight through your participation in this film project?
I feel like with our family specifically, I don't see a lot of families that utilize nursing and so forth. And I think that's a lot of because of Addie had a lot of really specialized and high-end, medical needs. But, I think there's a solution to everything. So, we try to be as normal as much as possible with whatever we have going on, and for us, nursing is part of trying to have that normal life, that allows her to be able to go to school, that has, having a dedicated nurse with her to go to school and, just doing all of that. I feel very much that, my husband and I, we really enjoy working full time, and being able to provide for our family and just having that sense of normalcy totally. I mean, it's not a normal situation, but it's just all in how you cope with it and come up with solutions that, fit how you need it to do. And, I think overall the documentary from our perspective, highlighted that portion of it.
Thank you, Amanda. Erika, Kassi, anything you'd like to share there?
For me, it's really about, for Elijah's story, is the late diagnosis he had and how to hopefully get more messaging out how critical it is for undiagnosed SDS patients invisibly waiting with all these symptoms, that the medical community can, get to a path for faster diagnosis. Because again, like I said, I try not to think of what could be, but you can't help but wonder, what if they would have diagnosed him sooner? Would he still be here with us because he was able to have that bone marrow transplant before the diagnosis of AML? Kassi, I don't know if you have
I think for me was the fact that SDS was not an end-all be-all. He still wanted to enjoy some of the normalcies of life, such as spending time with his kids, with his wife, with his parents. And he still was able to live a very full life, regardless of the diagnosis that he received.
Thank you. I know we're running a little short on time, so I did want to ask at least one other question. And my question to you is, so we know, again, our hope is that this is going to impact how researchers approach clinical trials as we're working for therapies, towards therapies, and you have all shared wonderfully what it means to be part of this community, for other families to get involved, those messages of hope, and thank you for that. But if you were to tell researchers just, one sentence of what is most important for you as an SDS mother, spouse, caregiver, what do you want researchers to know? The biggest thing. And it's a loaded question, but Amanda, we'll start with you, and then we'll go to Erika and Kassi.
Just the one thing is we do struggle. We have struggled through Addison's medical history of Shwachman-Diamond being recognized. So we went from our home hospital, which is a well-recognized hospital, that we had to figure out that rare was not recognized there. So we had to go elsewhere. So if I had one thing, it would be that it would be much more well-known from the start, as Elijah's mom said there, if it could be from the start. I think Addison's, even with Addison, if I didn't have to battle all of that with her home hospital, I think that we could have had some different outcomes as well on our end. But it's a battle to have these physicians understand and to listen. So on top of knowing, having it just more well known would be helpful, so that we could get appropriate treatments and get the ball rolling on a cure.
Thank you, Amanda. Erika, Kassi?
I think early detection is key, and resources. The Charlotte, we're from North Carolina, and the big hospital in Charlotte really mistreated Elijah's cancer approach, was completely mismatched to what he really needed as a SDS patient, because they didn't know he had it. And he really suffered for it. And just more, the resources, we have some wonderful resources. It's just, it doesn't seem to me on this side that doctors have enough forthwith to think to go to them in time. And I mean, over 100 doctors, we were told, examined his case in Charlotte and couldn't get to the bottom of this. And genetic testing wasn't even really in that mix until after a meeting of 100 doctors. That just blows my mind. So early detection and really encouraging them to get into genetic testing as soon as possible, for me, is what I think.
I agree with both Amanda and Erika. If we had been able to detect it earlier, I think he could have possibly lived a more full life, gotten to spend time. You know, his son was only a year and a half old when he passed. And so he could have, you know, watched him grow, watched his daughter grow. But we just didn't know if they could just, you know, provide early testing, provide more ways to help those with SDS in terms of treatments and things like that.
Thank you all three. A great, you know, early detection, awareness around genetic testing, all of that. It is important. And thank you for being so willing, again, to share. We really look forward to seeing you in this film. We're really excited. Like Eszter said, chills. And with that, I will pass it over to Eszter, and thank you again.
Thank you.
Thank you. Thank you, everyone. I wish we could have another hour to just talk, but at the very end of the meeting, we'll have an open coffee chat, so we can continue these conversations like a family. So please stick around at the end. And with that, we'll jump right into session 2, back to a more cerebral kind of approach by thinking about research and drug development. So session 2 is called the Cosmos of Research. Next, please. So we named this session a Cosmos because there is a whole range of different types of research that are necessary to get an idea of a possible approach to treat a disease into actual patients. So it's not linear. It's not like making it in the lab work and then all of a sudden it becomes a drug. There's a lot of, a wide range of research that is necessary to build the infrastructure, the knowledge, the insights, the connections, the community to make that happen. For example, we often hear about new drug development, phase 1, 2, 3 clinical trials. And our next speaker, Juliane Mills, will introduce us to the details of that. But there's also other approaches. The preclinical, the lab work is also important. We need to understand the disease as a whole in order to understand what to expect, what to measure, what we need to treat. So these would be natural history studies, for example, and registries. And then there is another often underappreciated area of research, which is called qualitative research, which is asking more open-ended questions to get to the root cause, what actually matters to patients, for example, so that follow on more specific questions can be asked down the road. Next, please. So with that, it is my pleasure to introduce Juliane Mills as our next speaker. She's an ally in the rare disease community and to drug companies developing therapies for rare indications. Juliane's work is about connecting the dots between cutting-edge science and the everyday lives of those affected by rare conditions. She's not just a researcher, she's a champion for better health, working tirelessly to ensure that new life-changing treatments move from the lab bench to the patient bedside as smoothly, as swiftly as possible. And I had the great privilege to work with Juliane over the years in different settings, and I'm going to hand over the mic to her.
Thanks very much, Eszter. Thanks to everybody for your time today. It's a real honor and a privilege to be here with you. In designing clinical trials, my role is to work with drug developers to make sure that we get the data that we need as quickly and accurately as possible, while also balancing how we do that with how patients live their everyday lives. So Amanda, hearing you talk about the nursing that you need and the multiple specialties that you had to visit, trying to find a way to do clinical trials so that you can bolt it on to a visit that you're already doing, or that we can provide compensation to make it easier to miss a day out of work. Those are the kinds of things that I try to build into the trials that I design. If we go to the next slide, just a reminder, I'm not a doctor. Don't take anything I say here as gospel. Please talk to a medically trained professional. If we go to the next slide, please. So like any other industry, medicine and pharmaceuticals love to use their own language and acronyms. So I want to begin today by making sure that we're all aligned by what we mean when we talk about clinical trials. And in my presentation today, I'm referring to it as an experiment that we do to learn if an intervention is safe in humans and if it works or is effective in treating a disease. And an intervention here can mean testing a new medicine, like a drug that's just been invented, which is what I'll be talking about today. But we also do clinical trials to learn if a medicine that has already been approved in one disease might work in another disease. That's called drug repurposing, and we'll hear about that later as well. Regardless, all clinical trials follow a plan. We refer to that plan as a protocol, and everyone involved in the clinical trial, every doctor, every nurse, lab tech, and every patient needs to follow the protocol. The protocol describes all the parameters of the study and all of the steps in the trial, including who can participate by listing the inclusion and exclusion criteria, how many people can participate, what drugs will be given and for how long, and how the safety of the people who volunteer for the trial will be monitored to make sure that we're kept safe every step of the way. Next slide, please. There are many reasons why somebody might choose to participate in a clinical trial. The most common ones are the ones I've listed here. There are no treatments for your disease, or there are treatments, but they don't really work for your version of the disease, or they have side effects that makes it difficult to take them. Or you come from some sort of altruistic purpose that you want to help move science and improve the lives of others. And it can be any of these, none of these, all of these, whatever moves you to think about a clinical trial. If we go to the next slide, there are certain stages that every clinical trial, generally speaking, follows. It's the same stepwise process that we go through to learn if products are safe for humans and if they work. The first step is the preclinical phase, and we'll hear more about that from Eszter in a minute. This phase can take a long time, and it's all done in the lab. None of it is done in humans. It involves those things you heard about earlier, the cell lines, the animal models. And once researchers doing that work have a drug that they think might work, they can submit a plan to the FDA to get permission to test their drug in humans. Only when that permission is granted can researchers move on to the next phase, which is phase 1. So each phase of clinical research is designed to manage the risk of a new medicine against any possible benefits. And so we go through that calculation at each step and each phase of the trial. In phase 1, we don't know a lot about how humans will react to a new drug. So phase 1 is only done in a few people, usually only healthy people, and they only get one or a few doses of the medicine. If the results from a phase 1 trial show that the medicine appears safe, then we can move on to phase 2. In phase 2, again, we're looking for safety, but we start in phase 2 to look for signs that show that the medicine might work. If phase 2 shows that, again, it continues to be safe and that the medicine might work, then we can move on to phase 3. And after phase 3, all the data are collected from preclinical through phase 1, phase 2, all the studies that were done. They're submitted to the regulatory agency, and because I'm in the US, I'm talking about the FDA here, and they decide if the medicine is safe and effective, and then they decide if it'll be approved for sale. And so you can see in each phase, because we know more about the medicine, the risk can be a little bit lower, so we can include more patients at every phase. This is difficult to do in rare disease trials because there aren't a lot of patients to begin with. So it may be a little bit different for clinical trials, but this is generally the process that we follow. If we go to the next slide, if you've never participated in a clinical trial, you may wonder what happens. And I'm going to speak very generally here, right? But generally, there are about 5 steps: pre-screening, consent, screening, treatment, and follow-up. Pre-screening is when a doctor or a nurse would have a general conversation with a patient or a patient's family about a specific trial to see if they might be interested in participating. They might look at the patient's medical records to see if they meet the criteria that are outlined in the protocol that specify who can and who cannot participate. And if it looks like the patient might be a good candidate for the trial, then the patient and the family will be formally invited to participate in the trial through a much longer conversation with the doctor before being asked to sign a consent form. This consent form will outline all of the expectations from the patient for participating in the trial, all of the things that will happen, all of the tests that will be done, the drugs that will be administered, and how long the patient can expect those visits to last, and how long they can expect the trial to last. Once the family, a patient signs the consent form, then we can start the formal screening process. And in this screening process, the patient may go through very specific tests and assessments like blood tests or x-rays to see if the patient meets all of the inclusion criteria and none of the exclusion criteria so that they can participate in the trial. Something to note, even if you just had a blood test, you may have to have the same blood test done again because it needs to be done in the context and under the control of the clinical trial. So I hear that oftentimes from families that it can be frustrating because we just had this test done. There's a rationale for why it's being repeated for the clinical trial. Sometimes you can reuse the data, but not always. Before a patient can get any drug, they have to complete the consent and screening step. And again, that screening step is really important because if the patient doesn't meet all of the criteria in the screening step, then they cannot participate in the trial. Once you've passed the consent and screening steps, then the next step is the treatment phase. And this is when a patient will receive the trial medication or a placebo, if it's a placebo-controlled trial, and potentially have tests done during their trial visits. That treatment period can last a short amount of time, can last years. And then eventually you move to the last step in the trial, and this is the follow-up step. In this step, generally speaking, the patient does not receive any medications, but is only monitored for a period of time. Again, may continue to have some tests done to make sure that the patient is still safe, see if there's any delayed effects of the medication, and if there were changes as a result of the medication to see how long those changes might last. So generally speaking, those are about the 5 phases that we'll see in a clinical trial. We go to the next slide. I want to talk a little bit about how trials are done in other countries. Again, in the US, I'm talking about the FDA, but clinical trials can be done in almost every country, and I have run trials in many different countries outside of the US. Each country has a regulatory, a safety, and ethical oversight of the clinical trial process. Even some of the sites, the hospitals where trials are conducted, may have their own safety and ethical review. In the US, we refer to that regulatory agency as the Food and Drug Administration. In Europe, it's referred to as the EMA or European Medicines Agency. And in Japan, it's referred to as the PMDA or Pharmaceuticals and Medical Devices Agency. But even in countries that I haven't mentioned, there is some regulatory oversight that defines what needs to be done to keep patients safe and to make sure that they are treated ethically. The next question that I often get is why aren't tests, if we go back to the quick, thank you, why aren't clinical trials available in every country? And there's competing interests here, right? So drug companies need to make money, and that means they need to do their trials quickly and cheaply. So they need to go where the patients are because recruitment and enrollment in a clinical trial typically is a very long part of the clinical trial process and can be very expensive. So if we can do recruitment and enrollment quicker, like making studies easier for people to participate in, then that helps our bottom line overall. So we look at things like the prevalence of the condition, and For example, if there's a genetic founder effect in Italy and we know somebody there who's been treating several patients, the drug company may only decide to do the trial in Italy. We also look at where are the experts who treat patients with these diseases, because we know that they know patients and might be able to enroll them in the trial. And so we might set up trial sites in the countries where there are disease experts. We also look at the timeline. So every regulatory agency, including the ones that I mentioned, goes through their ethics and safety review process, and from country to country, that process can take a different amount of time. The institutions, the hospitals, also sometimes go through a safety and ethical review. And sometimes those reviews are done consecutively after the regulatory review, and sometimes they can be done in parallel. And it varies from country to country, but all of those things impact the timeline before we can even start enrolling patients in the trial. And so depending on how long that process takes and where the patients are and who can treat them drives how we pick countries. We may also pick countries based on what's available in the market or market competition. If a drug is approved and available in the US and lots of patients are taking it, then there's not a high unmet need for patients to participate in a clinical trial. And we may look at countries where that product isn't available or it isn't approved, because we know there are patients who need another option like a clinical trial. And so we may decide to move the trial into those countries. So there's a lot of different variables that go into that decision. It depends on the disease, it depends on the market, it depends on the country. So these things are generally weighed against each other. And lastly, if we go to the next slide, then let me just wrap up with some resources. If you want to learn more about clinical trials, I am always available to you as a resource. If you want to learn more about clinical trials, making trials accessible to families and patients is my passion. And so if you have ideas about how to do that, or questions about what is a clinical trial or how do you find out more about them, there are some links here that will answer that. But again, I'm always a resource to you and happy to answer any questions. In repurposing, on what phase does the clinical trial begin? Great question, and ascent. So let me take the last one first, ascent. So different countries have different definitions of the age of majority. Here in the US, we typically refer to that as 18. In some other countries, that's 17. Could be as low as 16. In those countries, we need to get assent from those patients as well as consent from their legally authorized representative. And so in that case, there's a consenting and assenting process that goes through. And spoiler alert, if that patient who is under the age of majority is still in the trial once they achieve the age of majority, they need to reconsent then to be part of the study, because now they have the authority to speak for their own choices and they have to sign that form again. And repurposing, what phase does the trial begin? So that depends on whether the product has already gone through to approval for a medication. If that's the case, then depending on how similar the patient populations are between the disease in which it was approved and the disease in which we want to test it, we may be able to skip phase 1 and maybe do a shorter phase 2 study. If there is no similarity, like it was an adult approval and we want to go into pediatric patients, then the regulatory agencies may ask for additional safety data at the preclinical stage, or they may ask for a phase 1. So it really depends on the type of disease and patient population in which it was approved and who we're targeting to use it in a new purpose. All right. Is there anything I missed? Otherwise, I'll give it back to you.
No, on my end, your last sentence froze, but I think the rest of us probably heard it. I just wanted to end also, if like the formulation significantly changes, that might also require a repeat of phase 1, like if it's a very different dose or if it's liquid versus something else. So it's complicated, but the goal absolutely with drug repurposing is to save time and money and skip as many of the steps as possible while keeping the patient's safety at front and center.
Exactly.
Any other questions from the audience? I think that was the only one I saw. So thank you. Thank you so much for the wonderful overview, Juliane. And so let me jump right to the next topic. So there's this great website resource by the US government that they put together to just gather information about clinical trials. So most of the clinical trials that Juliane mentioned will be listed on Clinicaltrials.gov. They try to make it a little bit more user-friendly, the language and the layout, but it is still pretty technical. However, if you want to know if there is any clinical trial for Shwachman-Diamond Syndrome or a particular symptom you're experiencing, it's definitely worth looking on Clinicaltrials.gov. And if there's something that interests you, bring it to your medical team and discuss with them whether it warrants further investigation or consideration. Next, please. So this is what it looks like. If you go to clinicaltrials.gov, you get a search window and you can enter your disease or condition, and then you'll get a whole bunch of options, list clinical trials that are listed. The US government doesn't endorse them. It's really just a catalog of information that the investigators or the trial sponsors submit to clinicaltrials.gov. Next. So one clinical trial that I wanted to mention to you all is kind of at the intersection of drug repurposing. So it's a drug that was developed for something else many years ago. It's called Mavorixafor, and X4 Pharma, a pharmaceutical company, has licensed it to repurpose for the treatment of WHIM syndrome, which is a rare genetic immune deficiency disorder. And they have received market authorization for that. So it now became a drug. And now the company is looking to see if other immune deficiencies or congenital neutropenias or other neutropenic conditions may benefit from the drug as well. So it's repurposing in that sense. And they are currently running a phase 3 clinical trial. So as Juliane mentioned, the safety phase 1 and 2 don't need to be repeated. And now they can test in certain population whether the drug has beneficial effects. Next slide, please. So X4 Pharma, the company, has shared with us some slides to show you if you want to look further into this trial. It's called Forward for Chronic Neutropenias. Next slide, please. And there is a link with more information and the link to the information on Clinicaltrials.gov as well. So all this information will be shared in our resource section of the study of the SDS-POPs meeting center. If you want to look further into it, talk to your physician or reach out to the company directly. We are not endorsing the drug or the trial in any shape or form. You're simply sharing information of research opportunities that are out there. Next, please. So next, I wanted to highlight yet another type of drug repurposing approach. This one is using artificial intelligence. So EveryCure is a nonprofit company founded by Dr. David Fajgenbaum. And the idea with this... Hello, sorry, I just got some technical issues there for a moment. But getting back to the overview of EveryCure, this is, I don't know exactly where you lost me, but basically this is a company founded by Dr. Fajgenbaum. It's called EveryCure. And the idea is that they take all the information that exists about existing approved drugs, about their mechanism of action, like what they do, how they work, and feed into that system also all that is known about diseases, like what pathways are affected in the cell, like is inflammation involved, that sort of thing. And then the artificial intelligence algorithm is going to look for a match. Is a certain drug likely to have a positive effect on that particular disease? So it's repurposing in that sense, that the drug was originally developed and approved for a different condition, and now the company is looking to see whether it could benefit additional patients with additional disorders. So Dr. Fajgenbaum is a physician scientist at the University of Pennsylvania and one of the youngest professors to receive tenure in the history of Penn medicine, co-founder and president of the Castleman's Disease Collaborative Research Network, and co-founder and president of EveryCure, that's the company I just described, and national best-selling author of Chasing My Cure, A Doctor's Race to Turn Hope into Action. So I mentioned before, if you're able to fill out the survey about our meeting today, the first 10 people who submit the form will receive a signed copy of this amazing book. It's really such a great read. I thoroughly enjoyed reading it. And with that, let's play the video that Dr. Fajgenbaum shared with us to share about his story and the journey of the company.
There were a lot of emotions when I was laying on my deathbed, all 5 times. I'll never forget what it was like to have my dad, my sisters, my friends coming in to hug me and to say their final goodbyes. And my doctors were sure that this was it. And I remember my sister sitting next to me and saying, "Just breathe, Dave, just breathe." And I remember hearing that, and I said, "You know what, I can do one more breath." And amazingly, that's when that first set of chemotherapy kicked in. My name's David Fajgenbaum, and I'm a physician scientist at the University of Pennsylvania. I'm also a patient with Castleman disease. Castleman's is this horrible rare disease where basically your immune system attacks and shuts down your vital organs. And it's just relentless. It doesn't stop until it kills you, or you use treatment to stop it. During the time when I was battling Castleman's and also in med school, it was just so tough because I was simultaneously receiving chemotherapy to try to keep my disease in remission. So I felt pretty horrible from the medicines I was taking. And then I was taking care of patients, and that's all I wanted to do. But I was also dealing with just fear of the unknown. I didn't know when it was going to come back, and it kept coming back. I nearly died 5 times in those 3 years. And so I had this clarity that, like, okay, if I want to survive, I've got to find a drug that can save my life. And I can't just sort of hope that some researcher somewhere is going to figure out. I've got to turn my hope into action. So my mission became really clear, and that was figure out what's going wrong in my immune system, and then figure out, is there a drug that was made for another disease that can maybe fix the problem in me, in my disease? So I started performing experiments in a lab just down the street on my own blood samples. I discovered that a key part of the immune system called mTOR, it's basically a communication line for immune cells to talk to one another, was turned into overdrive. And maybe if I use an mTOR inhibitor, I could turn this off, and then maybe that would keep my disease in remission. And so I talked to one of my doctors about this idea, and he basically was like, look, we got no other options for you, so why don't we try an mTOR inhibitor? And now I've got 10.5 years of remission while I'm on this drug. So rather than going on to do a medical residency, which was sort of always the plan, I went right to joining the faculty here at the University of Pennsylvania, where I could just dedicate 100% of my time to research. All I wanted to do is, after finding this drug that could save my life, is to get this drug and as many other drugs as I could to other people to save their lives. I joined together with my 2 co-founders, Grant Mitchell and Tracy Sikora. We launched EveryCure, and the premise of EveryCure and our mission and vision is to unlock the full potential of every FDA-approved drug to treat every disease in every patient that those FDA-approved drugs can possibly treat, not just the 1 or 2 diseases that those drugs were intended for. And so we've been talking about it, dreaming about it, but it really wasn't until the advent of next generation and more recent artificial intelligence technologies that have helped us to say, okay, we can scale what I do in my lab 1 disease at a time to look across every drug and every disease. One way to think about our AI platform is to think about the way that streaming services recommend different shows for you to watch. And the way that it does that is it uses something called a random forest algorithm. And the random forest algorithm will basically look at all the shows you've looked at in your past. And then it profiles all the shows out there that you haven't seen yet. And then it matches them together and gives you a score. We utilize an almost identical algorithm to make matches between drugs and diseases. So our algorithms characterize and come up with a profile for every drug and every disease, and then gives a score for how good of a match it is between that drug and that disease. Figuring out ways to leverage these incredible technologies so that way humans aren't spending days and days reviewing through every study when you could utilize generative AI to do that in a matter of seconds. I live with the belief that hope is really, really important, but hope is not enough. Hope has to inspire action. It's just been the honor of my lifetime to be able to work here at the University of Pennsylvania and through EveryCure to save lives with cures that are literally just sitting at the pharmacy shelf.
So this was really a beautiful example of another way of thinking about drug repurposing, really on a systems level. And I'm happy to share that I talked to Dr. Fajgenbaum to make sure that SDS is included in the algorithm, and we are. So hopefully we will learn more over the next couple of months to see if there is any drug that is worth looking into that could benefit our patients. So next up, I would like to introduce Dr. Bauer. So Dr. Bauer's work, gene therapy, currently is at a stage that is preclinical. So it's not ready to be used on humans. There's still significant work to be done on animal models and cells. And then, as Juliane described, once there is enough data available that the researchers are confident that it has a really good chance working in people, then the researchers would work with the FDA to get it approved to be tried out on patients in the future. And there's different mechanisms to do that. Dr. Bauer might mention that in his talk as well. This would be called an investigator-initiated clinical trial. And so Dr. Bauer is currently at a conference or flying back from a conference, but he was able to share with us a recording to share with you. Unfortunately, due to time constraints, we had to cut a few slides in the middle. Those slides were focused on his vast and in-depth experience with sickle cell disease, as his lab really played a very important role in past development of gene therapy for sickle cell disease. And we'll be happy to share the full recording with you offline or on the meeting platform, or maybe host him at a future webinar. But now I'm excited to share with you what he recorded for us. Oh, and I forgot to properly introduce him. Dr. Bauer is a principal investigator and staff physician at Dana-Farber and Boston Children's Cancer and Blood Disorders Center. He is assistant professor of pediatrics at Harvard Medical School, principal faculty at Harvard Stem Cell Institute, and associate member of the Broad Institute for MIT and Harvard. His honors have included American Society for Clinical Investigation, Young Physician Scientist Award, and a whole lot more that you can read later on. He's a very accomplished researcher in the field of gene therapy, and we are really excited and honored that he is now also passionate about Shwachman-Diamond Syndrome. And now off to the video.
Hello. My name is Daniel Bauer. I'm a pediatric hematologist and physician scientist at Boston Children's Hospital. I'm going to talk today about opportunities for therapeutic gene editing for Shwachman-Diamond syndrome. So in hematology, we actually already have a standard curative approach for blood disorders, and this is bone marrow transplant, and this is where we can replace the blood system of an individual who has a blood disorder by transplanting hematopoietic stem cells from a healthy donor. And these are the stem cells that make blood cells for life, and so this can replace the blood system of a person who has a blood disorder and cure their disease potentially. But this has challenges, and this depends on a donor pool that's limited. We need to find the best matched donors, and that can be difficult. And even with a matched donor, there remains some incompatibility between the immune systems of the donor and the recipient, and that leads to risks such as graft rejection and graft versus host disease that make the procedure more complex. And there is some morbidity and even mortality associated with this procedure. And our alternative approach could be to collect the hematopoietic stem cells from the patient, him or herself, to modify them to fix the problem of the blood disorder, and then to reinfuse those cells to replace the patient's blood system with a blood system that no longer has that blood disorder. And how can we do that? Well, we can take an approach known as genome editing. And here's an artistic depiction. And the idea is we can make precise changes in the DNA sequences that are associated with disease in living cells. And then we can fix the potential of those cells to restore health. And if we do that in hematopoietic stem cells for blood disorders, those are the cells that that produce the whole blood system, and they can overcome the blood disorder. And so how do genome editing technologies work? Well, there are these amazing molecular machines that have basically two parts. There's one part that's for DNA targeting, and you can imagine if you'd use an analogy of sending a package, this is like the address, the street address, and the zip code that gets that package to just the right place. And then there's the contents of the package, which are the DNA modifying parts that once the genome editing tool has gotten to just the right place in the genome, it can make a change. And if there's a mutation that's causing a blood disorder, they can potentially fix that mutation. And so genome editing today is based on engineered CRISPR systems. And CRISPR systems are actually naturally occurring immune defense systems that bacteria have to fight off viruses that infect those bacteria. And this is showing the structure of CRISPR protein wrapped around RNA, shown here in orange, and then DNA, shown here in blue. And so you can see how the RNA and the DNA make this tight connection. And here's kind of a cartoon version of this, where normally the DNA would be double-stranded and have this blue strand and gray strand. But now the RNA, shown here in orange, can invade and target just that specific sequence of DNA that matches the orange and blue match here. And now, once that locks in, the CRISPR protein can engage, and these two triangles show these cut sites where the CRISPR protein can make a change in the DNA, and that can be a permanent change with a one-time CRISPR exposure. And here's kind of an artistic vision of like the universe of DNA in the nucleus of a cell, and in a two-part recognition mechanism, the CRISPR system first finds these kind of yellow docking sequences, and then this red RNA sequence, if it matches the target DNA, it can unzip the DNA, zip in the RNA and allow the CRISPR molecule to engage and make a change in the DNA. And what's so powerful about these systems is just by changing the RNA sequence, we can retarget the CRISPR system to essentially anywhere in the genome, so we can target almost any DNA sequence we want to by making a simple modification of the design of these CRISPR systems. Now, when we think about gene editing, it's really important to think about the precision of the approach. We want to target where we want to target, but nowhere else in the genome. Imagine these are two chromosomes, a blue one and a gray one, and there's a, let's say, a place in the blue one we want to target, and this is like in green showing the intended edit, but we want to avoid all these unwanted possibilities, like some other edit at that region, or targeting another chromosome, or making a deletion, or a translocation of the chromosomes, or loss of chromosomes, or shuffling of chromosomes. So we need to be very precise when we're doing this to make just the changes we want to make and no other changes. And we need to deliver these molecular machines into cells, right? They're no good unless they're actually in the cells of the patient, where they can fix the problems associated with the disease. And so we have all sorts of ways to deliver these gene editing systems into cells. We can use electrical energy through electroporation. We can use physical means with mechanical poration of cells. And we can use biochemical and nanotechnology methods where we can engineer particles that can deliver CRISPR systems into cells. And so we have all these options available to us. We did extensive study of the precision of the approach. Without going into detail, we learned a lot along the way. And one of the key findings was that genetic variants that we all have that differ between us can create risks of off-target effects. And we found a common genetic variant that could produce an off-target potential for the initial version of the gene editing therapy. And so we use that information to design an even more precise therapy. And we developed a new method to look for these off-target effects that was very useful in allowing us to get FDA approval of our therapy, and I think is kind of a new standard in the field to look for genetic variant associated off-target potential of gene editing. So with all that background, what are we doing now? We're working on therapeutic gene editing for Shwachman-Diamond syndrome. So as you know, Shwachman-Diamond syndrome is a genetic condition where the blood disorder component is a main risk for morbidity and mortality, and that there's a mutation that almost all patients carry in the second intron of the SDS gene, which greatly reduces the splicing of the gene and the production of a healthy SDS gene. And why this is a favorable feature from the standpoint of gene editing is that we could design a universal editing strategy to fix this mutation that could then be applied to almost every patient with Shwachman-Diamond syndrome. In addition, the disease itself is associated with decreased fitness of the hematopoietic stem cells, and there's frequently compensatory somatic mutations that are observed, which suggests that a relatively low threshold of editing could provide great clinical benefit. And we have proof of concept studies underway, and we have very encouraging preliminary results that we can get great efficiency and precision of gene editing for Shwachman-Diamond syndrome. So our vision is to develop an investigator-initiated clinical trial of therapeutic gene editing for Shwachman-Diamond syndrome. And I should say this is a collaboration with my colleague, Dr. Akiko Shimamura, who's also at Boston Children's Hospital and who leads the SDS registry. And so our next steps are to complete our proof of concept studies, to do off-target analyses and safety studies, obtain clinical grade editing materials, develop our cell manufacturing process and our clinical protocol, all to achieve regulatory approval so we can initiate the clinical trial. And we have a track record of doing this, and we kind of know the next steps to proceed with. So how can you learn more about this? Well, you could visit the SDS registry page listed here. You can email me. I've shown my email address. And how could you help? Well, it would be terrific for our proof of concept studies to have more samples from SDS patients, so you could participate in the registry and share samples of blood, mobilized blood, and bone marrow. And if you'd like to donate to support our research, the way to do that would be through the SDSregistry.org. You can see the website with the donate button, and this is through the Boston Children's Hospital Trust. So thank you very much for your attention, and I'm really excited to further develop our approaches for therapeutic gene editing for SDS.
That was a great overview of gene therapy approaches for SDS, and really exciting to see that such experienced researchers are really looking at it. I know several other labs actually who are very invested into gene therapy for SDS, and the way we at the SDS Alliance are supporting this work is by investing in the development of mouse models, which will then accelerate taking all the learnings from the lab and being able to move it forward into a mammal, and then from there to apply into humans. So with that, I'm gonna move on to our next speaker. Our next speaker is Dr. Vanessa Merker, who is gonna share with us what qualitative research is and how it can improve health outcomes. I am a molecular biologist, so I was initially really used to lab work. But since trying to accelerate therapy development, I learned very quickly that it takes so much more, and so much more is needed to develop and know how to get treatments and better health outcomes of people. Dr. Merker is Assistant Professor of Neurology at Harvard Medical School and Massachusetts General Hospital, and the Director of Research for the Family Center for Neurofibromatosis and Schwannomatosis at MGH. Dr. Merker is trained as a health services researcher and works to improve healthcare delivery and research for people with rare diseases by leveraging information collected directly from patients and their family members. Specifically, she uses qualitative methods to understand patients' healthcare needs. She develops patient-reported outcome measures for use in clinical trials, and she engages patients as partners in the design and conduct of research. She recently joined our advisory board to share her expertise in these methods, and we are very grateful to have her speak to us today. Vanessa, it's all yours.
Thank you so much, and it's so great to be here. As Eszter said, I'm a little bit newer to the Shwachman-Diamond Syndrome community, but super glad to be here. I work primarily, my background is in research on neurofibromatosis and schwannomatosis, which are also cancer predisposition syndromes, so genetic diseases that predispose to different kinds of tumors. And it's really interesting to see the overlap in kind of some of the needs and scientific development between rare disease communities. So that's something I'm personally committed to, is, trying to see how we can improve care for rare diseases generally and not reinvent the wheel from each symptom to symptom. So I focus specifically in a type of research called qualitative research, and I'm super pumped to tell you a little bit more about kind of what that research is, especially as we gear up to start doing some qualitative research in Shwachman-Diamond Syndrome, which you'll hear about a little bit later in today's events. So just to get started, what is qualitative research and how is it different from traditional quantitative research? So really, qualitative research is a whole family of research methods that are really designed to get in-depth understanding of human experience and behaviors by collecting non-numerical data. So things that aren't just numbers, things like text and audio and video. And that's really so that we can focus on kind of why things happen, how things happen, more than just counting up what is happening. And so I'll talk a little bit about the different ways that we do this and involve folks. But this is really something that we want to go beyond just the numbers. Numbers are important in understanding how frequent things happen, but to really get behind, go behind the numbers, understand why they're happening and what people's experiences are really like. And so quantitative research, that is research with numbers and counting, is great when you already have a very clearly defined subject. You know exactly what you want to measure it. You know exactly how you can measure it in, say, a survey or with objective kind of assessments like MRIs or blood tests. And it's great when you want to know, precise numbers like 25% of people with SDS get this kind of complication. It's not so great, though, when you have subjects that are a little bit new, maybe not new to you as people living with SDS or having family members with SDS, but new to researchers, new to pharmaceutical companies, where we don't exactly know the best way to measure something, or something's just so complex, there's no way to distill it down into a single number. And that's where qualitative research really comes in to help us understand, trying to understand why things are happening. And to do that, we focus in on really a small number of issues or cases or people's stories, and then really go in depth to try and do this analysis. And the main distinction between kind of quantitative and qualitative research is whether it's kind of summarized in a number or summarized with words and descriptions. When we look for patterns or themes and kind of explanations that take, whole paragraphs to explain because they're so complex. And so the ways that we collect qualitative data, there's many ways, some that you might be asked to participate in or even have already participated in are individual interviews, so one-on-one interviews in which a person a researcher comes to you maybe on Zoom, maybe in person to ask questions about things like what it was like to try to get a diagnosis of SDS for your child, or what it's been like on a clinical trial, whether a drug has been working or not, and kind of what side effects you're experiencing. You might have also qualitative surveys. So many survey questions that you get are quantitative. They're ultimately summed up with a number, but you've probably seen open text fields, those kind of boxes that say, can you please share anything else that you would like or any other information you have, where you type in those sentences. That's also qualitative data in its own way, because we're looking to see not just to summarize it, how many people said X or Y, but to really understand the reasons behind the other numbers or responses in the survey. There are also focus group discussions, which would be like interviews, but would be a group setting. So typically there would be maybe 4 to 8 people and one researcher all talking together about questions. That way you guys can bounce ideas off of each other, and we can kind of, even though we can't go as in-depth into each person's story, we can have everyone talking and sharing and kind of coming up with ideas. And then there's other ways that we might, I think, less commonly do this, but this says record keeping, but I would say more document analysis. So looking at people's medical records or looking at social media posts that they write about their experience, or discussion boards. If you've ever had a discussion board or something like a Reddit group, things like that can even be some of the data that we can use for people who would rather, type out their experiences, or who already have documents that we can analyze, like medical records. And another type would be observations. So this comes from anthropology and the anthropological kind of background, but oftentimes we can learn a lot as an outsider researcher kind of watching medical processes. So maybe coming to the inpatient hospital, watching what the doctors and nurses do and talk about to try and figure out ways to optimize that process or make it go better. And all of this kind of labor in doing this data collection is in service of really trying to illuminate your experiences, your perceptions, your attitudes, kind of how are people behaving, how are patients and doctors communicating and interacting, and how can we improve that? And I have this picture here. It's a kind of like an illustration of some people seeing the world glass half empty, glass half full. But I use it to show, there's a lot of complexity into all of our feelings and emotions and what's happening. And lots of people could perceive even very similar things different ways. And the only way you would know that is kind of by talking to them. So really the heart of qualitative research is talking directly to people who are experiencing things, patients, family members, frontline doctors, to see what is happening and how we can hopefully make it better in the future. And I know that Eszter had talked about the Food and Drug Administration's patient-focused drug development program, but I wanted to come back to it to say that it's also been a big impetus for doing more of this qualitative research and really trying to highlight patient stories, because the FDA, in trying to regulate new drugs, has really realized that we need to follow these systematic approaches to getting patients' experiences, figuring out their needs, what are their priorities, and really trying to incorporate that into the whole drug development cycle. And so that means even at the very beginning, before we even necessarily have drug treatments or other therapies, really trying to understand what are the impacts of the disease that matter the most to patients. How could we measure them in a clinical trial to see if they're improving over time? How can we tailor clinical trials to help meet the needs of patients, make it easier to participate? And then all the way even after we have treatments, how do we communicate the benefit to patients, to doctors, to insurance companies who are making decisions about what to cover? And so that's why I liked this slide that actually comes from a research company that helps do qualitative interviews embedded in clinical trials. So as part of a clinical trial where you're getting a new treatment, also having interviewers who check in with you at the beginning and middle and end of treatment to see how things are going, and all the different ways that this data is just so helpful, both to us as researchers learning more about the disease, but also to pharmaceutical companies and regulators like the FDA trying to make decisions about what therapies to develop. And that really helps us in so many ways, but including figuring out what's the natural history of the disease. Without treatment, what was it like? What was happening? What were patients' journeys? Where are they going to in the healthcare system? We can know a list of what people's symptoms are, but what's really their needs and priorities? Which things would they tackle first if you had limited kind of bandwidth to tackle different things? In terms of methods, this can help us figure out how are we going to measure these same symptoms or issues in a clinical trial? How are we going to show that this treatment improved that over time? And we also want to know how meaningful are the outcomes that we see. In one of the diseases that I work in, a common metric for our trials is that a tumor shrinks 20% or more. So there's a numerical outcome that we're going for. But then there's the question, what does that mean? How did you know 20% should be the threshold? What about 50%? What about 10%? And so you need that qualitative contextual data to say, well, once we get to 20% or more, that's starting to really make a difference in how people can function. That's making a difference in their day-to-day lives. And that's what we really need to show, because we need to put the numbers in that context of people's lives of how this is actually making a meaningful change to say, hey, this drug or treatment is actually, worth it. It's not just going to change a number on a test or an MRI. It's actually going to change how you function day to day. And another thing we're exploring is how to use qualitative data to think about benefits and risks. Everyone has different thresholds for what kind of benefit risk trade-offs they're willing to make. what side effects of treatment would you accept in order to get a really big benefit in a different area? And being able to explore that helps us target, what treatments might be the best for different people. And overall, we're just hoping to get, use qualitative data in tandem with quantitative data about new therapies to really just get a holistic view of what is it like to be in this trial and what is it like to be someone with SDS or other rare diseases. And just as an example, I'll show, since we haven't done it qualitative research in SDS yet, but it's coming, I thought maybe it would be interesting to just see what is it kind of like, what do people learn from this? So this is in a different disease called Merkel cell carcinoma, so another cancer, where they did interviews with people interviewed in a clinical trial. And so this is some of the kind of data, not that you have to read every specific box, but really this helped researchers figure out this timeline. What is it like before you get diagnosed with cancer, and then the diagnostic process? What are the limitations of current treatments? What are the side effects that are really bothering people that we'd like to avoid in new treatment? And when you sign up for new treatment, what is it that people are expecting? What would be a success? What would be something that people aren't willing to trade off? And so coming together to put this impact or sort of information together about people's treatment journeys and diagnostic journeys is actually really helpful to us as researchers and to clinicians to get this view of kind of what was happening outside of the clinic, what's happening in people's everyday lives. And this is just another picture from the same trial, but this was how qualitative data they used to help understand what are the areas of people's quality of life that are actually changing. It's easy numerically to say that some people have improved disease, which is the first row, or stable disease, or worsened disease, which is the last row. But you can see these quotes are really what give this context to are people improving? When we ask them about, let's say, their physical well-being, their psychological well-being, is it getting better over time? Is it staying the same? And so you can see some of these quotes which help kind of analyze how we understand what people are doing and whether they, in their own day-to-day lives and in their own impression, are getting better or worse. And just to wrap up, I'll share this quote. This is from someone I interviewed who has the disease NF, one of the tumor predisposition syndromes I talked about, and I interviewed them in a trial. And they had said, I've said that my chief criticism of Dr. X is that he treats me like a pile of symptoms and not a person. That can be really, really frustrating. That's why I'm really pleased that you're doing this additional qualitative research arm. I think there's a lot more to be understood about NF from a great deal of perspectives that aren't currently in the medical literature. And even though this is a quote about NF, I think it applies to SDS as well, in that, it's only been fairly recently that we've really embraced listening to patients directly as part of the drug development process is not an aside, but something that's really critical into understanding what people are going through and effectively treating it. And so I hope that if you have the opportunity to participate in some qualitative research, you'll think about joining, because we really learned so much from the people who are actually experiencing it. So with that, I'll end. If anyone has questions, I'm happy to take them, but otherwise we can keep going.
Thank you so much, Vanessa. I've been looking at the chat and I don't see a specific question for you, but I'm just so excited to be able to share later on today a qualitative research project that we are about to launch. So stay tuned. And now, sorry. And now I'm excited to move on to the next speaker, Dr. Sharon Savage, who will teach us about natural history studies. Dr. Savage is the director of the clinical genetics branch and clinical director of the Division of Cancer Epidemiology and Genetics at the NIH. Her research program combines clinical, genetic, epidemiological studies to advance the understanding of cancer, etiology, and improve the lives of individuals with complex cancer-prone disorders. Her work in inherited bone marrow failure syndromes, SDS is one of those diseases, has discovered numerous new genetic etiologies and quantified the complex clinical manifestations faced by affected individuals. And it's my honor to hand over the microphone to Dr. Savage.
Wonderful. Thank you so much for having me. I do apologize if in advance if my Wi-Fi signal isn't great. So if I'm freezing, I will turn off my camera. So again, thanks for having me. It's great to be here and talking with families with SDS. And I thought I would. I'm thinking about what a natural history study is. I have some examples to show, and I thought I'd tell you just briefly about the clinical genetics branch at the National Cancer Institute. So in our branch, we really are trying to understand the causes of cancer. So in the case of something like SDS, we really want to understand why do people get SDS, and if they what's the genetic cause, what's the biology, how does that specific variant affect the actual biology of the patient? But then also we really try to work towards assessing how living with that disease affects them and their family. Are there things we can do to make it better for them, to reduce symptoms, to reduce cancer risk, et cetera? And then even though I study rare diseases, in the inherited bone marrow failure syndromes, we're also thinking about it at the context of the population. So what can we learn about the biology and what it's like to live with one of these rare disorders? And how can we use that to also help us understand the causes of cancer across the whole spectrum of people? So a natural history of disease, this is a definition from the National Center for Advancing Translational Sciences. So first I wanted to just define the natural history of a disease, which is basically the course a disease takes in the absence of intervention in individuals who have the disease, from either the time it starts until it goes away or until the person dies. So in the setting of SDS, it's the natural history of it is basically starting at birth, finding out that the child has these symptoms, and then knowing what happened over that person's lifetime. When we talk about a natural history study, it's a pre-planned observational study intended to track the course of the disease. It seeks to identify demographic, genetic, environmental, and other variables that correlate with the disease's development and outcomes. So thinking about SDS, it might be thinking after we define the gene that is causing SDS in that specific patient, we find that variant, what else is going on in that patient, and how does that correlate with the gene that is affected in them, and how is that also related to maybe their family history? A natural history study could also include patients who are receiving the current standard of care or emergent care, which could alter the manifestations of a disease. So it's really kind of you take someone starting today and you follow them, and you are very specific in what you're following and the data you're collecting, but you're not intervening. So if you need a treatment, you get the treatment and you keep being watched and followed. That's what that means. So this is a slide that maybe some of you, if you had the opportunity to meet Dr. Alter when she was, before she retired from the NCI, this was a comic that she liked to share that was made by a colleague's son. And welcome to the clinical genetics branch. Lots of questionnaires. And this is really key for natural history studies and including epidemiology studies. So in that, taking that broad-based spectrum approach to understanding cancer causes, disease causes, and the outcomes for patients, at the NCI, we have a spectrum of rare diseases that our investigators study. Our clinical genetics branch has 12 different principal investigators or scientists who are really focused on different aspects of the causes of cancer. And some of those folks are focused on rare cancer-prone disorders, including things you may have heard of, including Levi syndrome, Fanconi anemia, the other bone marrow failure syndromes. And the approach I'm going to talk about is really a natural history approach, which is what we apply to all of these, including work that was done with Dr. Alter, Dr. McReynolds, and with Ashley Thompson, who I know is on the call, in SDS as well. So in all of these studies, when we're studying a family and when we're looking at a rare genetic disease, and this goes for all rare genetic diseases, I think, when folks are designing studies, is we want to understand what's having this inherited disorder. So what are the complications and problems a person with SDS has versus what's just part of being a member of that family, right? We all know that certain disorders run in families in different ways. So how much of that might be SDS? How much of that might be because they have a, say, grandfather who has a history of heart disease? So our approach to studies at the NIH really start with the patient or their family members, where they either call us, or now we have a new online system and an electronic enrollment opportunity. And we basically, they fill out a screening questionnaire, which asks some very basic questions. There's a referral nurse who reviews that and presents it to the study team, which is the doctors and scientists and nurses and research assistants who are all working on the study. The study team says, yes, maybe they're eligible, or no, they're not. Maybe we need more information. If they're eligible, in our natural history studies, we then send questionnaires. And some of these questionnaires can be quite lengthy. Again, we start with family history because we want to know what's part of being a member of that family and what's part of just having this specific illness. And then we also do really detailed individual history questionnaires, and we also assess medical records. Any records that folks want to share, we abstract and put into a database. Now, the way our study works is that all of those folks become part of what we call the field cohort. That means they've signed consent, they're fully enrolled, they're full participants in our study. We help them and we help their home physicians do whatever is needed for the optimal care for that person, but we just haven't actually seen them at the NIH Clinical Center. Some patients even allow us to help us actually by having samples sent to us. So they'll add on a blood draw if they're going for a regular checkup, and they'll send us extra tubes of blood or send a saliva sample, that sort of thing as well. For our study, a subset of individuals do come to the clinical center at the NIH, which is a full-fledged hospital. It's a research hospital. And those folks become part of what we call our clinic cohort, where they are evaluated by subspecialists and the specific study team. So this, I think, is pretty similar to a lot of the approach for natural history studies that other investigators also use. And some of you may know about our inherited bone marrow failure syndrome study. Shwachman-Diamond Syndrome, of course, is one of those bone marrow failure syndromes. And this was launched by Dr. Alter, who's shown here in the red shirt, in 2002. And Dr. Neelam Giri, who some of you may also know here, has been the principal investigator since Dr. Alter retired 2 years ago. And I'm going to actually talk more specifically about dyskeratosis congenita and the telomere biology disorders. But we basically took this approach because when we look at the scientific literature, people like to report interesting cases, right? So a physician or a scientist will report, oh, I have this patient with this rare disease and they have this rare complication. So that's a publication that gets out into the scientific literature. But what doesn't get published is I have a patient with a rare disease and they have not developed all these other things, right? Because you sort of only publish and focus on the complications. So what Dr. Alter really asked from the very beginning in 2002, he said, well, we know people with these inherited bone marrow failure syndromes have a whole spectrum of complications. They have bone marrow failure, thinking of SDS, failure to thrive, right? Bone abnormalities, risks of cancer. but we didn't really know what those numbers were. What is an actual person's risk when we look at it from an epidemiology perspective? And so that's why we launched this study, and we've been following these patients, and we continue to follow them even now. So I'm just going to give you an example of dyskeratosis congenita because it's one of the diseases that I study the most. And I wanted to walk you through this because I think it also gives a really good example of the value of natural history studies. So just very briefly, DC, which I will call it, is a marrow failure syndrome. Patients have high risks of bone marrow failure, but they also have these skin abnormalities. They have nail dystrophy and these white spots in the mouth called leukoplakia. Now, the very first publications about dyskeratosis congenita were well over 100 years ago. And at that time, everyone thought it was X-linked, meaning that it was only boys were affected. The moms were healthy. They pass on the X chromosome to their son, and the son is affected. But what happens was over time, as we learned more, female cases were reported. And then eventually, it was really noted that it wasn't just X-linked, that there were autosomal dominant cases, there were autosomal recessive inheritance patterns, and then some individuals actually had no family history, but their clinical complications were all very similar and were consistent with the same disease. So the first gene discovered is called dyskerin, this red gene here, and it was the first link to telomere biology. And telomeres are the caps at the end of your chromosomes that help keep our genes and our genetic material stable. So one of the first things that Dr. Alter and the team did in our DC component of the marrow failure syndrome study was work with collaborators to create a diagnostic test that was not genetic. Because again, 20 years ago, we did not know all of the genes that caused the disorder. The genes that had been identified only accounted for about a quarter of the patients. So understanding the biology, we've measured telomere length, found that actually measuring telomere length is a really good diagnostic test. And this was by following patients. So again, this was with about 10 years of follow-up from the initial start of the study. We were able to publish that the robustness of telomere length as a diagnostic test. So then we said, what about my genes? And in this case, we mean the other kind of genes, right? Your genetic material. So by following patients in the dyskeratosis congenita cohort, what we've ended up actually is advancing the field so that now we no longer call it dyskeratosis congenita in and of itself. It's really recognized that this is a spectrum of telomere biology disorders. So I first started working on this disorder in 2006. I discovered TNF2 as a cause of disease in 2008. At that time, we were up to maybe we knew the gene in about 30-40% of people. And then I just put this up because over time, what you can see is that because we've kept following patients, genetic technology has changed over time. We've been able to get more and more genetic causes identified. And now we know that in this spectrum of diseases, there are probably at least 18 different genes that can be affected and cause very similar disorders. We didn't know that 20 years ago, and we also wouldn't know that if we weren't doing that careful clinical characterization that we started all that time ago in the natural history study. And then I'm just going to show you this data real quick because I think it also shows the importance of this long-term follow-up. So when we first published the risks of cancer in people with TBDs, we compared their risks of cancer with that of the general population through an NCI registry. And what you can see is here, the observed to expected, the patients had we estimated their risk of cancer was about 11 times higher than in the general population. The risk of tongue cancer at the time, we estimated around 1,100 times higher. That's really, really high, but the number is small. We had 66 patients in this study. 9 years later, we published on 197 patients, and you can see the risks is still really high, but it's about 4 times higher than the general population, and we're starting to get more precise estimates of the specific risks of cancer. And now most recently, we have a paper that's under revision that actually we, for the first time, have been able to look at cancer in the TBDs by gene and start to adjust our risk estimates by the specific gene that is affected in the person. So you could imagine that all of this long-term follow-up has really helped us counsel the patients as to their risks, because as you know, not everyone is the same. And the more data we have, the better we can get at counseling our individual patients for their disease risk. And so through this whole big long-term follow-up of the TBDs, we've also identified a whole host of other complications that had not really been quantified. And this is just a busy picture to show that it's a complicated disorder, but now we really are understanding it more. And similar to with SDS Alliance, I work very closely with Team Telomere, which again, building on this these advances from the natural history studies, we now have clinical guidelines, and we've been able to help guide families better because we have more data. And that's really the gist. I think, you know, natural history studies, they are really, really important because you need to know, the way I see it is that you need to know why things happen and what is happening before you can actually intervene and work to make things better. And so that's where in rare diseases, these types of studies, I think, are really informative and really changing the course of the diseases and improving the lives of patients and families too. And thank you very much. I'm happy to take questions if there was time.
Thank you, Sharon. Any questions, please type them in the chat. I've been looking and haven't seen any specific question for you at this time. But thank you for the presentation and putting into context how much we can actually learn and advance the field without actually putting any new treatments in people. And in fact, learning about what's going on without putting new treatments into people is the basis of being able to tell what future therapies work, how they work, and whether they work and have a benefit to the patient. So thank you so much for the great presentation. If there are more questions coming up later, I'll forward them to you. And now it's time to move on to the next, not only the next presentation, but the next session. We are running a little behind schedule, but I am a softie and cannot cut anybody off because I really appreciate everybody's contribution so much. So bear with me. I will cut actually some of my slides. So welcome to session 3. This session is focused around research that we are collaborating on. Next, please. So coming back to our figure from our publication that I mentioned at the very beginning, our goal is to accelerate therapy development for our children. And there is a whole range of efforts and infrastructure that we are looking to develop further or implement if they're not available yet. Next, please. So as such, is model systems. It's really difficult or next to impossible to develop something from the lab and advance them into people without having the opportunity to try it on another mammal first, such as a mouse model or even cells from patients. So model systems are really, really important to get lab results and help develop them into medicines that can help people. Next, please. So at the very beginning of the SDS Alliance journey in 2020, we started conversations with the research community to see what kind of mouse model is missing and what is it that they need. And so we embarked on this adventure to develop a humanized mouse model. What that means is that we wanted to take the human gene, the human SDS gene, or SBDS gene, or a section as big as possible that's representative of the disease and put it into a mouse. And we partnered with the Jackson lab to get that work done. And then the plan was or is to, once we have the model mice available, that we would then work with SDS specialist researchers to characterize how SDS shows up in the mouse models. Next, please. So as you all know, all who knows me, knows that this project has taken longer than expected. In 2021, that's when the official launch happened. And we shared a great webinar with all of you. I was planning to show it here, but in the interest of time, we will skip it and share it as a resource later. The team, the JAX team, started to work on the actual work, making the mice that summer. And then by early 2022, we figured out that the mice don't seem to be viable. They tried it in a different strain too, that's supposed to be more resilient. And unfortunately, even in that strain, introducing the human gene sequence into the mice did not yield any viable mice. So we shifted focus on really investigating why that is and how can we overcome this problem. So we sat down with SDS experts and mouse model development experts to come up with strategies to first to understand what's happening and then how to overcome them. And the strategies we came up with is to reduce or increase the amount of other proteins that interact with our proteins of interest. And this can be done by crossing the mice with mice that have less of those proteins or more of those proteins to, in effect, tweak how much effective functioning, working protein there is of the SBDS protein. So what we need to achieve is to reduce the amount of SBDS gene product or protein to just the right level. So just the right level means just enough to cause some problems in the body, but not enough to make it not viable. So this work is currently still ongoing. As you can imagine, crossing mice takes time. They have offspring. The offspring has to be mated and characterized, and we have to have enough mice to get the data that we need to understand what's happening. In the meantime, when this was happening, we also learned that Dr. Bauer's lab also developed a similar or tried to develop a very similar model. The difference being is that they took a shorter sequence of the human gene that they put into mice, and their mice too were not viable. But we invited Dr. Bauer's team to have a discussion with the JAX team to figure out what's different, what's the same, and whether we can work together to make it work somehow, whether with his model or with the JAX model, and to learn from each other. And so that's where we are. So Dr. Bauer has generously offered his strains to the JAX team. They have been imported. This whole process of importing a mouse strain into JAX is also something that takes a lot of time, but it's all done. And so currently, the JAX team is trying these strategies. And so I'm very glad that we have our researcher from JAX joining us. Maybe before I hand over the mic, Steve, would you like me to talk a little bit about splicing, or would you like to cover it?
I'm happy to talk about it. I haven't given a deep dive into the splicing abnormalities that accompany the mutation for SBDS, but if this has been covered at some level at some point in the...
Yeah, I can just...
Go for it. Yeah,
I can go over it real quick then.
No
yeah. Jeff, if you can share my slides again. So basically, let's see, maybe next slide. Let's see where I put those slides about yeah, let's do the next one. So yes, perfect. So just for the audience, a quick refresher of molecular biology 101. So everybody is now familiar with what DNA is. DNA is what contains all our genetic information in the cell. It's this fancy double helix. The information that's encoded in there, what the cell needs to do is read that and make a protein based on the information in the DNA. And this happens in several steps. First, the DNA is copied and made into an RNA. But this first version of the RNA is too long. There's sequences in there that should make it into the protein and sequences that are just junk or not used for the protein. And the process to remove those non-coding regions of the RNA, they're called introns, to remove those, that process is called splicing. So splicing is the removal of the sequences of RNA that we don't need to make the protein. And then once those sections are removed by splicing, we end up with a much shorter RNA, and that's what's called a messenger RNA. That's what's always all over the news with the vaccines and everything. But that's this short messenger RNA is what the ribosomes read and then make protein based on the information encoded in the messenger RNA. In SDS specifically, the mutation that Dr. Bauer also touched upon, there is a mutation called the splice site mutation right at the end of exon 2 that almost all SDS patients have. So that particular mutation, almost all patients have that one. And that's really great for research or for drug development, because if somebody develops a drug that targets that area, most SDS patients might be able to benefit from that. Unfortunately, it's a tricky one because what that mutation does is it impacts how efficiently this non-coding region is spliced out, removed. So what we learned, and what Steve might touch upon, is that we know a fair amount of how splicing happens in humans and what gene sequences work better than others. That information is also available for mice. But when we put a human sequence into mice, then it gets tricky. Then it's really difficult to predict how well things will work. And basically, we just have to try and see what happens. And that's what we did. And we learned quite a bit. And I think now I can hand over the mic to Steve.
Sure, great. Thanks, Eszter. So what I will do is share my screen now. It was a great introduction.
I also forget to officially introduce you that you
That's
are
all right.
an investigator and our partner on the Jackson side to run this project.
Great. Thanks, Eszter. So I'll even take the time to introduce Jax a little bit to you, to the group as well. So let me make sure I get this in presentation mode. Can you see my slides okay?
Yes.
All right, great, terrific. So I'm happy. Thank you, Eszter, for the invitation, and also the introduction as to sort of the challenges and the ongoing work that we're doing to try to make a SDS model that actually recapitulates the key elements of the disease phenotype. As you'll see in a moment, we've been successful, as you and as has Dan Bauer, as you mentioned earlier, in the molecular aspect of this. We were able to engineer what we need to engineer. The challenge, of course, in the long run is making it have the validity that you need for ongoing preclinical work. So let me start by introducing the Jackson Lab overall. For those of you who don't know who we are, we're a nonprofit research institution based primarily, although not exclusively, in Bar Harbor, Maine. And we have a long-standing history of using mouse models as a way to understand genetics and biology and the biology of human disease in a lot of different areas and programs. We have also a long-standing tradition of education. We have a really well-regarded summer program where many Nobel laureates have spent their youth and their summer in a beautiful place right by the ocean and spent time with our scientists here at JAX doing research. But one of the ways we're best recognized is as a purveyor of resources for the wider scientific community. And this includes what I would say is our commercial end, which is selling existing mouse models, but we also have a number of large resource development programs. I've just listed a few here. These names don't mean anything to folks here, of course, but the one I wanted to highlight is what we call the JAX Center for Precision Genetics, which has a specific mission in building new models of rare and ultra-rare disease that don't currently exist and that are not available to the community. And so that's the place we hope to fill in. Just to give a little more intro, we have 2 locations. I mentioned our flagship location, our starting location in Bar Harbor, Maine, which is currently has about 1,700 or 1,800 employees, so it's actually quite large for our region. But we've also opened another institute within JAX, which is in Farmington, Connecticut, really close to the UConn Health Center there. And our expertise of the different investigators at each location are widely overlapping, but we also support each other in places where perhaps we're complementary as well. So for example, neurobiology is a big strength of JAX Bar Harbor, but not necessarily at JAX Genomic Medicine, although this changes over time with the different investigators we have. Okay, so the JAX Center of Precision Genetics. This is an NIH-funded program that the mission is to build and characterize mouse models of rare disease with a view towards preclinical adaptation of those models, while also providing a tool to understand disease mechanisms and pathobiology. So really how we work is we actually start with conversations with foundations and researchers who identify individual patient mutations or those mutations that perhaps have been identified some time, who nominate those that disease and those specific mutations be engineered at JAX for a mouse model to be built. And so that's where Eszter came to us and with the SDS Alliance to say we need this model. While doing this, I think, Eszter, you alluded to this quite nicely, we think about the potential genetic therapies that might be applied to this. We know we're in really a new world, as you heard from Dan Bauer, about using genetic medicine specifically to correct mutations. And so even though we don't know if those therapies are going to work out, when we engineer our model, we think about what do we need to do in order to support that kind of work, in particular humanizing some element or a lot of that gene. We also think about, so then of course, we engineer these mouse models. We also have the capability and have in some cases engineered human-induced pluripotent stem cell lines that could be used in concert with the mouse models. And then, of course, where it really gets interesting is when we do the characterization of those models. And ultimately, we begin, if feasible, testing potential therapeutic strategies, such as you heard from Dan Bauer, in support of a investigational new drug along the way. So this is kind of the way our pipeline works overall. Now, obviously, not everything makes it to this final stage, but we certainly hope they would when we start our projects. And as I mentioned, there's several of these programs funded, but what makes JAX unique in many ways is the fact that the vast majority of our nominators actually come from foundations that are very much like the SDS Alliance. So these are just a list of some, not even all, of the different foundations that we work with. And this, I think, reflects the fact that we are not a medical center. We don't actually have a medical center component to our research infrastructure at JAX. And so we're a place that many of you or many of the foundations have begun to hear, we're a place to come make a mouse model. And so so we get contacted directly by a lot of these groups, and really are thankful for the efforts of the different foundations to nucleate a research team around this where we can contribute specifically our expertise. So, okay, so just a couple other things about our center. I mentioned, JAX has this history of mouse models, but one of our real big strengths is our ability to engineer a wide variety of types of models within the mouse using fairly complex means to add quite sophisticated new elements to each mouse model. So for example, large-scale humanization, we can make structural variants that in the past were more difficult to make. We also have a huge capacity. So over the past 3-year period, this is slightly out of date, but I think the numbers are pretty similar. We've had over 2,000 projects to engineer mouse models for 400 different groups around the world, including 1,000 for a program that was structured to knock out every gene in the mouse genome. So a lot of capacity and a lot of expertise that's been built up along this. And then something that, Eszter alluded to in trying different strains of mice. We have a lot of different strains. Each one is a different genetic context, just like each person has a different genetic context. We've engineered on many of these backgrounds as well. And so we have that capability that's actually unique to JAX as well. And then from a phenotyping or characterization perspective, we've built this center several years ago now that is essentially a mouse clinic. It allows us to test different phenotypes or traits that each mouse model might have that might be relevant to the disease and do it in a systematic way and we hope a cost-effective way. But it allows us to sort of centralize all of those capabilities rather than being in individual professors' labs. It's a core service that our center can tap into in order to get the broadest array of characterizations for any new mouse model that we make. Okay, and so I think Eszter spoke a bit about this. What was the goal of our program? Our near-term goal was to make sure we could engineer this precise model carrying, this is just the nomenclature of the splicing mutation that the vast majority of patients with SDS have, into the mouse SBDS gene. And then we want to include humanization, and this is to facilitate these genetic therapies, not only the gene editing that you heard about, but possibly antisense oligonucleotide and these other ways that have really grown as potential approaches for precision therapy. We want to make sure we could support those in as much as was feasible. And finally, sort of where really the important thing is determine if our phenotype, that is the characteristics of the mouse, actually recapitulates the disease characteristics, and with a focus obviously on bone marrow failure and the hematopoietic phenotypes as well. So in addition to other phenotypes, but that's the one that was certainly most important. So where do we stand? Oops, it's not letting me advance. Let me try this. There we go. So here's just a schematic of how we approach this. So I mentioned, so Eszter sort of gave you the schematic of the gene. I'm only including the first 3 exons here. What we're showing you is what we built. So the gray rectangles are the exons that are mouse, and the blue represents areas of the gene that we've humanized. That is, we've replaced the mouse version with the human version. And as you can see, in doing so, we've also inserted that specific splice site mutation to 258 plus 2, which means it's plus 2 bases beyond the end of the exon, and it's a C to T mutation. And so this was actually the easy part. So I mentioned we have all the really great expertise, and so building the molecular mouse itself was pretty straightforward. But then as we delved into the function of this, we ran into some unexpected results. So what we expected was this, is that we insert this mutation, and then from what we know from the human disease, we expected this to trigger the use of a different splice site, so the different end to this exon, which would then go into into exon 3. And what would happen is this creates a stop codon. So this is what arrests the gene. And what oh, sorry, I've messed up my my animations, but what you can see here is what we expected is that between 90 and 99%, the vast majority of the gene would now go into this form, which is leading to a halted product, so a non-functional product. And what we expected is something between 1 and 10%. It was not clear, and then actually it seems to vary within the literature how much is remaining in people. But we'd expect some level of normal protein to still be produced as a result of that. However, when we bred the mice, as you might have heard, hold on, I have to advance here. So this is how we did it. We took the female mice carrying one copy of this engineered mutation and a male mouse carrying one copy of the mutation, and we looked to see if we could put them together. And, oh, am I seeing this properly? Oh, there we go. I can't actually see it because of the pictures here. What we found is that these were, as Eszter alluded to at the beginning, embryonic lethal. And what we further learned by doing what we call time matings, we can figure out when they're dying during gestation. The JAX model was dying very, very early in gestation. Actually, as we've since learned, significantly earlier than the Bauer model. And this is possibly relevant, or could be important coming up. So what is happening? So this is our expected function. What we've come to do, a lot of molecular analyses, but I don't want to bore you with, but what we found is the actual function is that for reasons we don't understand, is that, but reasons that there is some evidence this is occurring in people as well to a significant degree, is that this mutation not only is causing this strange splicing event, but in mice at least, it's actually primarily causing a skipping of this exon completely. Unfortunately, this results in the same stop codon, the same out-of-frame disruption of the gene, and we're getting 100% of this. The Dan Bauer's model, possibly because they only humanized this portion here, actually a much smaller portion than that, a very small portion in this region, does see some residual splicing according to this expected outcome, but not enough to rescue it to be viable. So we're a little bit stuck at this point. So what are we doing now? So as Eszter mentioned, we know well, I was going to say we know a little bit. We actually know a lot about what SBS is doing functionally in the cell, and it's part of this complex with this gene EFL1, or this protein EFL1, and this other protein EIF6, and the purpose is to trigger the release of this from the ribosome so that the ribosome can form and do what it needs to do in terms of translation. And so one of the ways in talking with Dr. Alan Warren and others is to, sorry, I'm moving this around so I can see my screen. He had generated some genetic evidence that by modulating the levels of these different components, you could see phenotypes, you can affect the phenotypes of SBDS mutants in different forms. So we had two hypotheses. One was that we could, if we reduce the amount of EIF6, that is the protein that has to get released, we might be able to rescue that lethality because maybe you don't need as much SBDS to get the job done. Maybe EFL1 alone could get that job done. We also hypothesized that if we reduce the levels of EFL1, maybe we can get away with just one copy of the mutant SBDS and still see a phenotype. So those were the two sort of ideas we had. And so these crosses, as Eszter alluded to, are now underway with both the JAX and the Bauer models. I will say the Bauer model, I said it here, it's more promising. I predict it's more promising simply because that lethality is later and there is some residual SBDS protein being made that we don't see in our JAX model. For whatever reason, our full humanization has led to complete skipping. So I would predict we're more likely to have success with that. And what we do know right now is that creating the double SBDS EFL1 mutant, they're alive. We have them, and we're currently characterizing their blood phenotypes at this point right now. So that's exciting. So that's underway. And we do know so far removing EIF6 for the JAX model does not rescue the lethality, which is what we were hopeful it could, but it is kind of what we expected. We are close to having data on the Bauer model. So I think within this year, we'll certainly have all the data to tell us whether these strategies are going to be successful or not. And so just to sort of summarize and tell you what our plans are, we're going to continue, as I said, evaluate the effects of these genetic combinations. And then the other thing we've talked about, although this is based on just speculative, trying to understand why we see these exons skipping, can we modify the junction between 1 and 2 to reduce that skipping? It raises the question, though, if you've done something artificial, how relevant is the model? So these are ongoing conversations. And there may be other ways that we've not discussed at this point, but could be considered to adding back just a little bit of normal SBDS to rescue the phenotype while still retaining the specific mutation that then could be addressed with genetic modifications, such as what Dan Bauer had described to you, which is actually looking like it could be very exciting. So with that, I'll just thank you for your attention and thank the people who did the work, particularly Steve Nealand, who runs sort of the first part of our model production group. Kat Lutz's my fellow PI, our funding, and happy to get any suggestions along the way. This is one of the cases where we wish we could get them. The engineering part came very easily, but the getting the mouse to work the way we want is still a work in progress, and we're going to keep banging at this. So thanks, and I'll take any questions if there's time.
Thank you so much, Steve. Audience, if there's any question, please type them in the chat. So far, I haven't seen any specific ones from you, Steve. Feel free to stick around or drop off. And because we are going to move on to the next topic very swiftly. So we are running about a half hour behind schedule. So what I'll do is I will move things around so that some of the topics I'll cover at the end so that our speakers who are lined up can come on closer to the expected time. So to wrap up this mouse model section, this model that we just heard from Steve about is to make the mouse genetically as similar as possible to a human SDS patient. So all the mouse's cell would have the exact same genetic mutation as humans have, and that type of model would allow a wide range of research, right? But as we learned over time through doing the work, is that it may or may not work as we expected. So in the meantime, we also started a second project to develop a very different type of mouse model that would be beneficial for additional types of research. Next slide, please. So I cannot tell you a whole lot of detail about it yet because we are still working out the terms with the collaborator. But the idea is to take cells, stem cells from a human patient and change them in a way that they become hematopoietic stem cells. And then mice, special mice that are immune deficient, can be transplanted with the human stem cells. So the mice would basically undergo a bone marrow transplant, but receive human stem cells, and in effect would then produce human blood or blood system, possibly even additional types of cells in their bone marrow, in the mouse bone marrow. So that this technology advances, this is all brand new, has never been tried for SDS patients before. So we are very excited to be able to build this new collaboration, and as soon as all the paperwork is figured out, we'll be happy to share more. So next slide, please. So again, back to the overall strategies that we are pursuing, back to our trusty little graphic. Next, please. So we want to contribute towards data and data infrastructure. And then other way of learning about the patient experience is look at the medical records. There are companies that collect medical records and information and make them available for research in a de-identified way. So names, contact information is not included, but all sorts of clinical information. So when doctors see a patient, they record what the encounter entailed, and all those data are coded with specific codes, and a lot can be learned from it. However, until last year, SDS didn't have a code, so it wasn't possible to associate those kinds of data with SDS patients in the past. We applied to get an ICD-10 code for SDS in the US, and it was successful. So we received the ICD-10 code in October of last year. So it's almost has been 1 full year. Next slide, please. So how that process was, it's all documented in our paper, and we have a dedicated web page around that. I will link that into the resources. But what I wanted to share this is, like, we got the code implemented October last year. What's next? How are doctors going to know that now there is a code? Well, the good news is that doctors who encounter SDS patients for the first time, their medical record software that they use will probably tell them, if they search for Shwachman-Diamond syndrome, it will pop up and tell them the code. But we were concerned that doctors who already have a lot of SDS patients, they knew that there was no code for SDS, so they may not have a reason to search their software for the code. And so we launched a campaign to inform experienced SDS physicians that now there is a code. We asked the patients to tell their providers about it, and we also reached out to many of the doctors we know. And then we were curious, well, did this work? So I would like to invite you guys to guess, right? So a year ago, nobody had the code because there was no code until almost a year ago. So we started with 0. How many patients do you think have, as of today, received our 61.02, our ICD-10 code? Please type your guesses into the chat. We'll just wait for a few seconds. All right, we have one guess. 86 is the first guess. Any other guesses? All right, 50, another guess, 50. We got 86, we got 50. Any other guesses? How many patients in the US have received the SDS-specific ICD-10 code over the past year? And drum roll, please. Next, oh, somebody says 40 something. All right. And next slide, drum roll, please. All right, so the answer is 515 patients. So 515 patients have received the SDS-specific code. So that is huge success, I would say, given how rare we think SDS is. So what this means, maybe SDS is not quite as rare as we think. And there's a lot to be learned from those medical records later on. We also learned that about 85% of the people who received the code received them this year and 25% last year. So that also suggests that our outreach efforts are also working. So back to our overview of the different focus areas we have. One is finding more patients, right? More data, more knowledge about the disease, more patients able to choose to participate in clinical trials or not. We need to identify as many patients as possible. And also, of course, having accurate diagnosis helps the patients themselves too. Or first and foremost, I should say, but also it is very important for research. Next slide, please. So we have a number of efforts along those lines. First, we often get asked about newborn screening advances. And there are basically two big programs that are looking to establish sequencing or genetic testing as a standard of care for newborn screening. Currently, newborn screening in most countries is not based on genetic sequencing, but rather biochemical assays. If you have kids, you have probably seen like the heel prick assays where they take a tiny bit of blood and then run some assays on that. But that can only cover so many genes, right? Or so many disorders. So in the UK, there is an effort to shift newborn screening towards a genomes-based approach, genetic testing-based approach. And there's a similar program in the US. At the time when it was launched, it was limited to certain hospitals in New York State, but I'm certain that it's going to be expanded more widely. So that study is called the Guardian study. So both of these are centered around using genetic testing or whole exome sequencing as the basis for newborn screening. In both cases, families have to opt in that they want to participate. And so we reached out to both of these programs to make sure that SDS is included in those panels. The Guardian study has confirmed that SDS is included on their panel. And when we reached out to the UK-based program on the left, initially SDS was not included. So we reached out to them and connected them with Dr. Alan Warren, who was able to connect with them and convince them to include SDS on those panels as well. And they have. So that's really great news for the UK community, is that if families opt in to this new form of newborn screening, they have a really good chance of getting a diagnosis early and without having to go through all the pains that most of us here on the call had to go through just in order to just arrive at a diagnosis. So I'll leave it at that in the interest of time. Next slide, please. Another area where we are investing in is to make access to genetic testing easier for patients. So when patients suspect that they might have SDS, maybe they are adult patients or the parents Google symptoms and they happen upon the idea that it could be SDS, they should approach their medical team to see if they could pursue a genetic testing for the disease. But sometimes the physicians are not able to help them getting genetic testing covered by insurance. And sometimes physicians are not even comfortable with ordering genetic testing. So for those cases, we have researched opportunities for free genetic testing. One of the partners we found is called Probably Genetic, and patients can self-refer, meaning they can reach out to this program, fill out a screening tool, and if they meet certain symptom criteria, they would then be eligible for free clinical grade whole exome sequencing based testing. So that's Probably Genetic. There's a link to the program on our genetic testing website. And then another program we are very excited to partner with is the Rare Genomes Project. based at the Broad Institute. And we'll have a speaker in just a moment about it. The idea here is that for some patients, even if they had whole exome sequencing done or other types of genetic testing, the results may still be inconclusive. They may not rule SDS in or out, and so patients might still be in the limbo whether it's SDS or not, or maybe they're just totally not able to access genetic testing at all. And so the Rare Genomes Project really developed a great program to help those patients. Unfortunately, both Probably Genetic and the Rare Genomes Project are currently only available for US patients only. But we have some other connections in other countries and opportunities to facilitate testing. So whatever your situation may be, if you need access to free genetic testing, please reach out to our team, genetics at SDSAlliance.com, .org, sorry, and we'll help you find the right resources. And with that, I would love to hand over to Mike to Melanie. I think, let's see my next slide. Yes, Melanie O'Leary. She's the principal clinical genomics specialist and the operations lead for the Rare Genomes Project at the Broad Institute of MIT and Harvard. And this whole genome-based research study aims to identify the genetic causes of diseases for undiagnosed families. Melanie, take it away.
Hi, thank you, Esther, and thank you to everybody at the Alliance and for everybody who's here to join us today. I wanted to talk a little bit more about the Rare Genomes Project research study that Esther was just talking about, which is where I've been working for the past 7 or 8 years, but relatively recently been connected with the SDS Alliance through shared funding through the Chan Zuckerberg Initiative and their Rare is One effort to try to help rare disease communities make science happen faster, quicker, as we're all searching for better things, better treatments, better interventions. So I'll talk for a few minutes and I'll hand it off to my colleague, I think Katie Russell, who is going to be talking right after me. The particular I am just a representative of a much bigger team that's comprised of clinicians, researchers, bioinformatics individuals, all different flavors of geneticists whose area of expertise is in analysis of genomic sequencing data and variant interpretation. And part of the funding that we received from the Chan Zuckerberg Initiative was to bring our genetic sequencing resources and make them available to members of the SDS community. Our particular project is a free and entirely remote research-based program where we perform whole genome sequencing to look for the genetic cause of all types of rare diseases, but including SDS. Our goals as a study are I think in line with the goals of many families in terms of we really want to increase access to families have to genomic sequencing services. We are a research study. We're not as fast as a clinical testing resource would be. So we always recommend that families pursue clinical testing when they can. But sometimes clinical testing is not available. Sometimes families don't have a provider who's feels comfortable ordering genetic testing, or some families still have a firm suspicion or a clinical diagnosis of a diagnosable condition, but just can't be diagnosed through the tests that they've had made available to them. And so we bring kind of the big sledgehammer whole genome sequencing to the families who enroll in our study. Our goal for individual families is to try to find the genetic basis of disease for them. Our goal for the scientific environment as a whole is to understand more about the genetic architecture that underlies lots of different types of genetic conditions to really help better understand genetically, what are the types of things that can lead to disease, and come up with better ways and tools and ways to make diagnoses for families. For individual families who participate, if we do identify a suspected genetic explanation for the symptoms that are being experienced in the family, we do have a return of results process where we clinically confirm the result and return it to families in conjunction with their local providers. A lot of people, I'm sure, found their way to this call because of having symptoms and a suspected, all the way probable or known diagnosis of SDS. Lots of genetic sequencing technologies exist. There's no one magic test that finds everything, but the three most common ones that are out and about right now range from targeted sequencing for individual genes or short lists of genes. If you Google genetic testing for SDS, sometimes you'll get a lab will offer testing for just the SBDS gene, or sometimes it's a short list of genes that also includes other SDS-related genetic causes, or genes that have diseases that have symptomatic overlap, where people can sometimes get misdiagnosed based on clinical symptoms. Whole exome sequencing is the next most broad test. It covers all of the known genes in our bodies and comprises about 1 to 2% of all of our genetic makeup. And then whole genome sequencing, I call it the sledgehammer, includes the whole thing. So it includes a small list of genes. It includes all of the genes, but it also includes all of the other connector DNA and regulatory elements of our genetic makeup. To be eligible to participate in our study, families have to be, they themselves have suspected SDS or other single gene disorder, or be a family member of somebody who does, and like Eszter had mentioned, currently live in the United States. Specifically with regard to SDS, the individual who is symptomatic has to have 2 or more of the following symptoms. We have a flyer. Eszter and the team at the Alliance has, it was a co-developed flyer, so they obviously have copies of it and can figure out how to distribute it, but there's a list of different symptoms here, and the criteria is just have to have 2 or more of the following. We don't accept everybody who applies to our study directly into the study. It does, all applications go through a vetting process. And I would say probably the most common reasons why we end up not accepting families into the study who at face value are eligible because they themselves have or have a family member who has SDS, are for people who come to us because they've had genetic testing in the past. They've had maybe 1 or 2 variants of uncertain significance identified, and what they really are looking for is someone to weigh in on whether or not those particular variants are actually disease-causing. We don't have other types of wet lab resources that could help lend credence or disprove particular variants of uncertain significance. So we're not the best place to help with that particular question. The other 2 circumstances where we do get applications that we would not accept would be if someone is a family member of someone who already has the genetic diagnosis in the family, or if someone who is seeking carrier testing. And that is because we do have finite resources, and our goal is to help as many families achieve a genetic diagnosis and rely on cheaper, easier to access clinical genetic testing for the carrier testing or for the testing of other affected family members. The process for families, we try to make it as simple as possible. So we do have an online application that is not too long, in my opinion, that can be submitted online directly or through one of our team members over the phone. We do not require that prior genetic testing have been performed, but we do encourage families when it's possible and accessible to pursue clinical testing first because it's quicker. For families who are accepted into our study, we do like to, when it's possible, enroll the person that has the symptoms, both of their biological parents, and any other family members who are also symptomatic. But participation by those family members is not required. We collect blood samples on everybody, drawn locally at a Quest Diagnostics or other blood draw facility of their choosing, and sent to us by FedEx, where we do submit it to the lab and submit for whole genome sequencing. We ask to collect medical records on symptomatic family members. And then hopefully on the back end, if we find a genetic diagnosis, we contact the family and work with them and a provider of their choosing to retest it and confirm the result in a clinical laboratory and return that back to the family. We're relatively early enough in the process where I don't believe we have any families who have indicated SDS as the reason why they've applied come spitting out on the back end where we've had data to analyze and a result potentially to return. But we are here, we are ready, and we're eager to work with any families who are in need. If you have any questions, you can let me know or let Eszter know. We're happy to give you more information. You're welcome to call us. And we just thank you for the opportunity to talk with you today.
Thank you, Melanie. Any questions for Melanie? I haven't seen any in the chat, but I'll happy to forward further questions as they come along. And next up is, can switch to the next topic. So one thing you touched upon, Melanie, is the VUSs. And so that's a big topic in itself. Well, VUS is a variant of unknown significance. So when patients undergo genetic testing or sequencing, sometimes the change in the sequence is known to cause disease. So that's what we call a mutation, and that can lead to a diagnosis. But oftentimes the change in the sequence is just a change, and we can't automatically tell whether that causes a problem or not. And so those are the dreaded variants of unknown significance. There's a lot of work around the globe with geneticists, and I don't even know what other experts are involved in this, but to get a handle on VUSs and what to do with them for clinical diagnosis. But all these variants also have relevance in just understanding how many patients there are for a certain disease. And so that's what's leading us to our next project I'd like to introduce, also in collaboration with the Broad Institute of MIT and Harvard, but a different group. So I'd like to introduce Katie Russell. And the title of the talk is Estimating Genetic Prevalence. Katie is a clinical genomic variant curator who researches and classifies genetic variants identified through GenomeAD, public databases, and rare disease genomics efforts. Prior to joining the Broad, she received her Bachelor of Science in Biology with a concentration of biomedical sciences and a minor in models and data from Trinity College in Connecticut. So this is really at the heart of genetics and sequences and data and what we can learn from it, especially as related to rare disease and SDS. So take it away, Katie.
All right, thank you so much. So I'm so happy to be here today. As Eszter said, I am Katie Russell, and I work at the Broad Institute as a variant curator. So I'm here today on behalf of the Rare Genomes Project and the Chan-Zuckerberg Initiative to tell you a little bit about the genetic prevalence study we did in EFL1. So EFL1 is another gene that can cause SDS. The main gene you usually probably hear about is SBDS, but I think Dr. Murray actually even referred to it in his presentation as well, that EFL1 variation in that gene can also cause this disease. So to jump right into what the prevalence study is, essentially the point of this study is to answer the question, how many of us are there? And the us we're referring to in this case are individuals with SDS due to variation in the EFL1 gene. And so I'm sure as a patient advocacy group, you already have a good idea of why it's important to know how many individuals are affected with a certain condition. But just as a reminder why it's important to know these numbers, it's great for reasons like patient recruitment. It's great for researchers to know when writing grants. It's also very important to know for pharma and biotech if you were to ever present about therapeutics. These are numbers that they like to know as well. So why do we do a genetic prevalence study? Well, typically when we think about trying to determine the prevalence of a disease or how many people have a disease, we often look towards counting. So, maybe it's counting how many people are involved in a group like this. Maybe it's counting how many people are reported in the literature to have variants. Maybe like Eszter referred to before, it's how many people have a certain ICD-10 code related to a disease. But we often know that that will miss people, that not everyone has, resources to be part of these studies, that not everyone with the disease gets included in these groups for whatever reason, and that there are probably more people with the disease out there that are not being accounted for. So to try and get a better estimate of how many people actually have SDS due to variation in the EFL1 gene, we're doing this genetic prevalence study. So instead of counting, what we're doing is we're using a mathematical model to estimate the proportion of the population that would have the causal genotype for this disease. So what I mean by a causal genotype is that because SDS is a recessive disease and EFL1 is a recessive gene, it means that a person would have two pathogenic variants in EFL1, one inherited from their biological mother and one inherited from their biological father. So how do we do this study? I'm just going to give a brief overview here, but the idea is that we're really putting together a bunch of resources that are actually pretty publicly available. We're not doing anything that involves proprietary data. We're just kind of combining a bunch of different resources. So what we do is we use what's known as a population database, and our team uses Nomad, which I'll talk to you a little bit more about on the next slide. But what we do is we look for pathogenic variants. So as Eszter talked about before, I'm a variant curator, so variants which are mutations in the gene get classified into different buckets depending on how much evidence there is for them causing diseases. So the highest level is pathogenic, which means they're very obviously disease-causing. There's lots of evidence that they cause disease. And then there are also those VUSs, which are ones that are a little bit more uncertain. We don't have enough evidence to tell either way if they're causing disease. So we look in Nomad and we look for those pathogenic variants, the ones that have evidence that they're causing disease. And then we're able to see how frequently they occur in the population and then use that frequency to do some math and determine this prevalence. So what is Nomad and why do we use it? Nomad is the genome aggregation database. It is also out of the Broad, created by a team we work very closely with. And essentially, it's supposed to just provide a snapshot of genetic variants floating around in the average general population. So right now, you can see in the bottom corner here, we have about 800,000 individuals' genetic data in it. It's all de-identified. We can't go through and pick out any individual in it. And again, it's supposed to be a snapshot of the average population. However, like many of these type of databases, it does still heavily skew towards individuals of European ancestry. Although with each update, we do try to include more and more individuals of non-European ancestry. And then what we're able to do with this database is look at how often carriers of variants in EFL1 occur. So you can see here on the bottom right, you can look at different types of variants and then how often we see individuals have these variants. But what's interesting is that this is not, people with SDS. This database is actually depleted for individuals with severe genetic diseases. So we're not seeing individuals with SDS in here. What we're seeing are the carriers. And by seeing how frequently those carriers occur in the population, we're able to do some of that math and figure out and really make an estimate of how many individuals we'd expect to see with SDS due to variation EFL1. And recently, just earlier this year, our team released Genie, which is the genetic prevalence estimator, which is a tool that allows groups like yours and other researchers to be able to kind of do this math of and to actually do the complicated math part of it. You're able to insert your gene of interest, look through a list of variants and pick out the variants that your group knows is pathogenic, usually with the help of like a genetic counselor. And then that should give you a number of the rough estimate that you can also kind of hone over time as you learn more about what pathogenic variants, what variants are pathogenic and what ones are VUSs. And so we were able to do this for EFL1. We returned just a few weeks ago our results to Eszter and Ashley. I know we're still working on a few of the details of exactly how and when we want to publish and share them. But it was a great collaboration. And yeah, thank you so much for having me here to speak and to talk about the project. And I'm happy to take any questions, or I'll put my email in the chat if anyone has any questions afterwards. Thank you.
Thank you, Katie. It was an amazing overview. I do see one question. In addition to genetic rarity, many practitioners are reluctant or unwilling to diagnose anything that would that would exist in the realm of zebras, let alone unicorns, therefore genetic testing is not performed. Yeah, I guess that just goes back to the challenges of accessing genetic testing. For this particular project, genetic testing is important so we know which variants are likely to be pathogenic. But luckily, many other parts of this work are actually not relying on genetic testing for a particular disease, but these are large databases of genomic sequences. So yeah, but it is a challenge on both sides, I think. Other than that, I don't see any questions or comments. If there are questions specifically for Katie, please type them in the chat. And if not, thank you so much, Katie. That was a wonderful overview, and we look forward to sharing more details about the findings soon.
Yes, thank you so much again for having me.
So back to my trusted slide here. This was a part of our efforts to find more patients, or at least know how many patients to expect and where to look for them, or how to think about prioritizing our approaches to find more patients. Next, we're gonna talk about, let's see, next slide. Oh, so next we're gonna talk about the more qualitative research that we are initiating. So this goes back to the idea that we want to share with regulators, such as the FDA, and with drug developers, such as drug development companies, what is it exactly that affects our patients the most, has the biggest impact on quality of life for our patients, what's most important to our patients, and we need a way to capture that information and communicate it to the right people in a way that they can act upon that those insights can inform a direction for therapy development or approval pathways. So I introduced Ashley actually at the very beginning already. She's part of our team now for about a year. And I let her introduce herself. And then new to our team for this particular project is Grace Lynch. I believe Ashley will introduce her too, but just as a little spoiler, she's a genetic counseling student at MGH. Yeah, I think I'll leave it up to you guys to wrap up the introductions and tell us about a new exciting project. Take it away.
Great. Thanks so much, Eszter. Let me go ahead and share my screen and we can take a look at these slides. All right, Grace, can you give me a thumbs up if you can see the slides? Okay, perfect. Well, wonderful. Thank you all for joining us here today. We are excited to introduce not only a new project opportunity, but also a new collaboration with the MGH Institute of Health Professions to you all. And so first I will give a little more background. We've got Grace Lynch here. She's a first-year genetic counseling student at the MGH Institute of Health Professions, and she will be joining me well, I'm a genetic counselor, but she will be joining me on this new project that we have to announce to you all today, titled the SDS Patient Lens Study, which is exploring the lived experiences, needs, and stories of SDS patients and caregivers. And so how did we get here? Eszter's already given some context to this, but just to give you a little background if you missed that earlier. So in summer of 2025, SDS Alliance, we are now slated to host an FDA externally led patient-focused drug development meeting. And we've already heard some about what these meetings are supposed to accomplish, both in Eszter's presentation and then in Dr. Merker's presentation. But just to remind you, the purpose of these meetings is to provide the FDA and other key stakeholders, such as product developers, clinicians, and researchers, an opportunity to hear perspectives from individuals with SDS about a variety of things, of which Dr. Merker outlined very eloquently earlier, but it also includes the health effects and daily impacts of SDS, experience with current treatments, and then factors influencing treatment selection. And so understanding the SDS patient perspective is critical in informing the FDA's decision during the drug development and approval processes. And so in preparation for this PFDD meeting in June, part of this is to take a look at what's available in the current medical literature. And so we did this and noticed that there's a gap in literature focused on describing the lived experiences of individuals with SDS. And so to help prepare for this meeting, SDS Alliance is conducting a research study of which I've already given you the title, the SDS Patient Lens, which is focused on investigating the lived experiences, needs, and stories of individuals with SDS and their caregivers. And so findings from this study will also inform future research, clinical care, and other advocacy initiatives in addition to the PFDD meeting. And so here's our patient lens study team. We are an interdisciplinary team across two organizations, both SDS Alliance and the MGH Institute of Health Professions. And so we have both myself and Eszter Hars from SDS Alliance on this team, as well as, and they've already introduced, Grace, the genetic counseling student at MGH, in addition to Dr. Merker, who you heard speak earlier, and another faculty member at MGH, Allison Serino, who is another genetic counselor and professor at MGH and their director of student research. So at this time, I will hand the slideshow off to Grace, and you'll get to hear a little bit more about the rest of our study.
Thanks, Ashley. Hi, everyone. Thanks for letting me join you today. As Ashley said, I am a first-year genetic counseling student at the Mass General Institute. And so I'm just going to give you a little bit more background about this project. So our research question is, what are the unique lived experiences, needs, challenges, and other stories of individuals with SDS, also including caretakers of individuals with SDS? So ultimately, we are focused on investigating the overall patient experience navigating the healthcare system in relation to a diagnosis of SDS in order to describe the diagnostic odyssey and patient journey. We also want to focus on learning about the social and economic impact of an SDS diagnosis in order to raise more awareness of the SDS patient experience. We want to identify research questions that are important to the community in order to have that kind of guide future research and advocacy efforts. And then lastly, we want patient perspectives on the current treatment options available. We want their input so that it can contribute to the future of patient care and therapy development. So a little more about what this study will look like. We are looking to interview individuals affected with SDS or caregivers like parents to children with SDS. We are aiming to interview around 10 to 16 participants, but we would love to talk to anyone who is interested, so the more the better. These will be conducted either via a phone call or video call. And they'll be semi-structured, meaning there's going to be some guiding questions, but we really do just want to give space for people to share their own experiences. And then this data will be used to kind of identify themes in order to not only prepare for that PFDD meeting in June, but also to guide future research, care, and advocacy. And then it is also important to note that all personal information and identifiers will be removed prior to analyzing and sharing data. So this data will be anonymous. So if you are interested in participating in this study, we are going to send out an email after this meeting in order to give you some more information and give you a chance to sign up. And really, we just want to hear from you. By participating in this study, you'll be contributing to foundational research critical to the process of drug development and FDA approval with the goal of future life-saving treatments for patients with SDS. So thank you for your consideration. We'd love to hear from you, and we're excited about this project.
Thank you. Thank you, both of you. As you can see, there is sun shining right in my face at the moment. We already got some interest in the chat, and I'm sure there'll be more interest coming once we share out these details. Any questions for Katie or Ashley right now that we would like to discuss? All right, so we are running behind schedule by a lot because I didn't have the heart to cut off anybody, and I'm so interested in everybody's expertise and insights. So if any of you, the audience, need to hop off or take a break, I completely understand and will not take it personally. However, we would still like to cover a few more things. This was meant to be the wrap up or the summary of the first 3 sessions. Basically, we invite you to step up and take action in any of the projects that we described today. We will send out a post-event survey, which will ask you questions about how the content was for you, whether it worked for you, and so on. And it will also ask whether you are interested in specific projects that we are running, including the LENS project you just heard about. Next week, or November 1st to the 7th, is going to be SDS Action Week, leading up to SDS Awareness Day on November 7th. And we're gonna suggest or invite you to participate in quick and easy actions throughout that week. So please keep an eye on those. A video I didn't have a chance to show you earlier due to time constraints is how we are actually going to prepare for the EL-PFDD meeting. We all have very important stories to share, but it will require a little bit of intention and work to think about how to most effectively deliver those messages, how to affect the impact that we want to achieve when we share our stories. And we engaged with a company specialized in helping us as individuals to develop those stories. The group is called Living Proof Advocacy. I might still be able to show you their welcome message at the end, but stay tuned for more information about that. So that will be part of preparing for the PFDD meeting. After this slide, I'm gonna go back and talk a little bit more about the GPS program. And then another thing coming up next week is our annual fundraiser, which is called A Million Steps Closer to Cure SDS. And this is really a fun kind of fundraiser where you can build teams in your community, and everybody can download a free app and count the steps they take during that week to add up to a grand total. Because as a community, we can probably surpass taking 1 million steps and get us closer to a cure. Also, while you're building teams, of course, this is a fundraiser. So if your team is able to fundraise and contribute towards our work, the more the better. So I'll send out some information about that too. It has been shared before in our newsletters and social media. And there's still time to join anytime. Actually, there's no preparation required. It starts on the 25th, October 25th to the very end of the month until Halloween. If you have other ideas for fundraisers, or if you have connections to companies that may be interested in sponsorship, please reach out. We are all ears. As a very small team, we really appreciate all the help we can get. And with that, the original plan was to switch after this to the coffee chat, but actually our time window has already closed. So I'm thinking actually I would like to go back and share with you some of our GPS work because I already have your attention, and we will do the coffee chat. If anybody still has energy to hang out afterwards, we can still chat. And then we also have a great video by a qualitative researcher who did research on how siblings feel about family life with SDS and what types of help or discussions have been helpful to them. So that we can also share still at the very end of this meeting. But now back to work, back to our work on GPS. So one of the areas that, again, back to our overview of what we do and why, we need as much data as we possibly can about SDS. Some of the data is lab-based, maybe lab values, or based in medical records, but a very important component that hasn't been systematically captured so far is patient-reported data. So these are stories, information, experiences that patients share directly themselves through surveys. Next slide, please. So in order to capture that very important information that's important for therapy development, and also to bridge the gap and let you all know about currently open clinical trials or research opportunities, we came up with this program called SDS-GPS. Next slide, please. So GPS stands for Global Patient Survey and Collaboration Platform. And really the sentiment here is destination cure driven by you. So we need your voices in order to drive research effectively. Next slide, please. The goal here is to give you unbiased access to information about relevant research opportunities and clinical trials. We as an organization are really, our goal is simply to let you know as much information as possible. And we don't really have a stake in whether you choose to participate or not. Our whole role is to just give you access to information. At the same time, we also want to give you the opportunity to have an impact on future research directions, and really to help patients now and in the future. We do this by really carefully listening at what you're telling us. And so we learn real insights about your lived experience when you share your experience through surveys. So with this information, we can then prioritize our programs and research focus to meet your needs best, communicate these needs to researchers, industry, and regulators so that future drug development efforts are actually addressing things that we care about. And it might also help with clinical trial design so that the clinical trials are set up in a way that we can actually participate, and may even inform clinical knowledge. So maybe we'll be able to share with clinicians what works for our patients, what doesn't, what types of symptoms are common, what isn't, that sort of thing. Next slide, please. So a very common question I get is like, is this a registry? Technically speaking, yes, it's a registry. It collects information about people with a certain health condition. But we don't call it a registry because really the goal here is to capture an aspect of life with SDS that hasn't been captured before and share it with as many people as possible who can help make life better for our patients. It's kind of like data in a rare disease. It's kind of like this image you might have heard about, the blind man and the elephant. If we all have blinders on, we are touching something, exploring something, or trying to learn about something, we will interpret information a certain way. We might think we are dealing with a wall, or we might think we're dealing with a rope or with a tree. But they all contribute, all this information contributes to really forming a complete picture and more understanding about the disease. None of this is in competition. Really, it's to enrich knowledge, foster collaboration, and sharing. That's why the SDS-GPS program was created. Next slide, please. The platform we chose for our surveys is called Matrix. You might see it in some of the following slides. So that's the platform. That's the tool that houses the consent forms, the data, the surveys, and it even provides productivity tools for you to manage your medical records or reports that you may want to share with doctors, or if you're traveling and have to visit an emergency room, you would have all your data accessible on your phone and don't have to lug around like folders and folders. Next slide, please. So this is what the program looks like on the Matrix platform. When you request access, you will choose whether you're a caregiver or a patient. Next slide, please. Then you have to agree to the Matrix terms of use. Next slide, please. And then you'll have the opportunity to add information about yourself as a caregiver or as a patient. If you sign up as a caregiver, you can then add one or several patients in the following steps. Next, please. You will be asked to enter some demographic information and how patient relates to you, whether you're a parent or a sibling. Next, please. And then for each participant, you will be asked to fill out an informed consent form. So any clinical research or any research involving people where data sharing or publication will be considered needs to go through a so-called informed consent form, which contains information about the study and is governed by an institutional review board. So an institutional review board is a group of bioethicists, if you will, who determine whether the study is set up in the best interest of patients. And only if they determine that it is, can the study proceed. And so this form specifically and our whole protocol for this entire project has been reviewed by an IRB and received green light to move forward. So for each participant, you will have to complete this informed consent form, which consists of just reading it, understanding it, and then signing it at the end. You don't have to fill out a lot of information there. Next. One of the questions will also include an invitation to upload the genetics report that has led to the diagnosis of the patient. This is optional, but very important for us to really understand how the patient's experience with SDS relates to their genes. And Ashley on our team reviews them on the back end to make sure that the information that is contained in there is really relevant to SDS. You'll also now have the option to opt in to receive information from us if you want to, if and when we learn that, for example, a variant of unknown significance has been reclassified, or whether if we learn something new about the genes and mutations and variants that appear in your report. If you want to hear from us about changes to those, we can inform you about those changes. Next. So this is an example of a survey. So this particular one is part of a survey that's SDS specific. We developed this particular one to learn more about aspects of SDS that are important to patients. Next. And then once you complete the first set of surveys, you'll have the option to get to this dashboard. Now we have a screen guiding you to where to go. On this particular screen, you can see all the surveys that you have already completed and all the ones that have been added to your account to be still completed. Next. So all this is really meant to be self-explanatory, and it guides you through the process, hopefully with ease. Most patients reported that it takes them anywhere from an half hour to an hour and a half to go through the whole process. And we are looking for you to share your story in this way about once a year. Currently, we have two types of surveys. Some are SDS specific and others are general in nature. So they go through questions. Do you have problems with your eyes, with your ears, with this and that? So they're general. Those questions don't mean that your patient is at risk for those complications, but it will help us get an overall understanding of the community, what types of issues come up most often or not at all. And we are looking to add a third type of survey very soon. These are called quality of life surveys. So they will ask questions such as depression, like has depression affected your ability to participate in work or school activities? Has pain affected your ability to participate in regular everyday activities? That type of question. So those types of surveys are coming soon, and we might ask them or ask you those questions maybe every quarter or every couple of months rather than just once a year. Next, please. So going back to this survey that I mentioned that are not SDS specific, asking questions from head to toe. That survey was developed by Clinigen for Genome Connect and is now used by many, many, many rare diseases, including Rare X, if you heard of them. That's through Global Genes and many other organizations. And using these surveys will not only allow us to get a broad overview of SDS in our community, but also be able to compare SDS to other rare diseases for cross-disease research. We invited the Clinigen team to share with us more information about these surveys and also how they relate to ClinVar. ClinVar being a big database that captures information about variants, genetic variants, and mutations. So Alexis Morgan is joining us from the Clinigen's Genome Connect patient registry. She has a degree in biology and in neuroscience and received her Master's of Science in Genetic Counseling from Rutgers University. She is a licensed certified genetic counselor at Geisinger for the past 3 years, where she works in both clinical and research settings. Alexis' research is with Genome Connect Clinigen Patient Registry. And after her talk, I can then show you what we already learned using these surveys from the GPS program. Okay, so stay tuned. So Jeff, if you can
Hello, my name is Alexis Borgen. I am a genetic counselor at Geisinger and help to coordinate ClinGen's patient data sharing efforts. Thank you all for allowing me to come speak to you. I am delighted to speak to you about Genome Connect. As you may know, over the last few decades, our ability to sequence someone's DNA has improved rapidly. While our ability to detect genetic changes or variants has improved over the years, there are still many questions around genetics and health, like what genes cause disease, what variants are tied to health concerns. The Clinical Genome Resource, or ClinGen, is an NIH-funded project that's dedicated to building an authoritative central resource that defines the clinical relevance of genes and variants for use in precision medicine and research. Ultimately, what this mission boils down to is that ClinGen hopes to understand what genes and variants are associated with disease and how genomic information can inform clinical care. ClinGen has a number of expert panels working to answer questions like, does this gene cause disease, developmental differences, and is this variant pathogenic? Those efforts to answer questions about genetic variants and their relationship to health rely on pooling genetic and health information together. As a result, ClinGen works with a number of stakeholders, including patients, clinicians, labs, and researchers, to pool data resources so that we're ready to use that information to answer some critical questions. Laboratories, researchers, and clinicians have been sharing genomic and health data for some time, but ClinGen felt strongly that patients can also serve as an important source of additional information. Patients are often experts about their own health history and can provide much more detail about their symptoms over time, diagnoses, and health. So ClinGen felt access to this additional health information could help increase our understanding of genes and genetic variants. So as a result, in 2014, the ClinGen team launched a patient registry called Genome Connect to empower patients to share their de-identified genetic and health information with publicly available databases. Genome Connect is open to anyone who has had genetic testing, regardless of results or diagnosis, so it is not disease or gene specific. Participants enroll online, consent to de-identified data sharing and recontact, provide their health history through patient surveys, and upload a copy of their genetic test results. Those data are then shared with publicly available databases. Genome Connect also partners with other registries to help them collect and share genetic and health data. Genome Connect works to collect health information that can increase our understanding of genetics and health. Genome Connect has developed health surveys that are used within Genome Connect and are shared with other registries like SDS. These surveys were developed in collaboration with some of the ClinGen experts. An initial set of survey questions inquires about general issues across the 17 broad categories or body systems, for example, bone marrow. Each question provides examples of issues in that system using patient-friendly language. Additional surveys can be assigned based on those initial questions. Currently, Genome Connect has 8 additional sub-surveys to capture information regarding specific health conditions participants answered yes to in the initial health survey, such as heart defects, arrhythmia, history of cancer. The SDS registry is using Genome Connect survey to collect genetic and health information. Although the survey is in patient-friendly language, on the back end, patients' responses are automatically mapped to human phenotype ontology terms. These terms are structured to enable data sharing outside of the registry. As more groups adopt these surveys, it allows for comparison across groups and across conditions because we are collecting data in a standard way. Genome Connect helps collect genetic information from participants in our registry and in other registries. Within Genome Connect, participants share their genetic test results by uploading the result or by providing permission for the registry to request their records from the laboratory that performed the testing or the healthcare provider that ordered it. Genetic counselors on our team review the report to ensure all of the genomic data is collected in a standardized way and assess its accuracy. Other registries like SDS can use Genome Connect strategies for reviewing reports to collect genetic information in a structured way. Within Genome Connect and some of the registries we work with, once genomic and health information are collected from patients in the registry, their de-identified information is shared with ClinVar, a publicly available database of genetic changes and their classifications. This database is where many clinical laboratories submit variants. Submitting this information to ClinVar makes the information publicly accessible to researchers, laboratories, and healthcare providers, so it can be used to answer questions like, is this gene tied to disease? Is this variant causative of health risks? And what symptoms are we seeing in people with these variants? This is an example of what a ClinVar record looks like. The top section is general information about the variant itself. And if you scroll down in the record, you'd see submissions around the variant. The submission highlighted is from Genome Connect. We submitted this variant on behalf of our participant, and the information is in there about how the variant was classified by the reporting laboratory, when it was classified, the inheritance pattern being de novo, and the clinical features of the participant are listed in structured terms. As you see, no identifying information is shared. So why is patient-centered data sharing important? Patients participating in Genome Connect or other registries can provide additional phenotype information regarding inheritance of the variant, and the ability to recontact participants for additional information as needed. Knowing patient-specific symptoms, inheritance of the variant, and if other variants were identified can help increase our understanding of genes and variants, which ones cause disease, and provide more specific information about genetic conditions. Patients enrolling in Genome Connect also provide new genetic changes that have not been submitted to ClinVar yet. From our submissions as of this year, 18% of genetic changes were not submitted by the reporting laboratory or another submitter. Putting this information out there in the public space can be useful for the next time a genetic variant is identified or similar variants are found in other patients. To illustrate how patient data sharing is helpful, I wanted to share a case example. I will call this participant Ella. Ella is a pediatric patient with a history of seizures beginning in infancy. She had exome sequencing ordered back in 2015, and a likely pathogenic variant in SCN8A, a gene associated with the seizure condition, was found. Her parents were tested, and the variant was not found to be in either of them, suggesting the change is de novo and unique to Ella. Because this genetic change has not been seen in healthy individuals, as de novo in Ella, and was expected to impact the protein, the clinical lab classified it as likely pathogenic. Ella and her family have an answer for her seizures and chose to participate in Genome Connect. We submitted Ella's de-identified genetic information to ClinVar. At the time of our submission, there were 2 other records in ClinVar. Lab 1, being Ella's reporting laboratory, had already submitted the variant as likely pathogenic, and Lab 2, another testing laboratory, had submitted it as uncertain. The Lab 1 record contained fewer details about Ella's clinical history and her parents' testing. Laboratories have these details, but often do not share them with ClinVar. After our submission, Lab 2 reached out to our team to ask more about Ella and confirm the variant was de novo. In their patient, they were unable to perform parental testing, and knowing if a genetic change is de novo can be incredibly helpful in understanding if a variant is disease-causing. We confirmed the information by re-reviewing Ella's report, which prompted Lab 2 to reclassify the variant and update their patient's report. Ella's results and care were not impacted, but her family taking the time to share her results ultimately enabled another family to get clearer genetic test results. In addition to helping increase our understanding of genetics and health, patient-centered data sharing can enable additional research opportunities for patients. By putting information into ClinVar, it helps researchers become aware of the registry and can lead to future collaborations and additional research. Registries can also learn about genetic test result updates and share those back with participants. For example, when a Genome Connect participant shares their genetic report, it might say the variant is uncertain. But when we share with ClinVar, we may see the variant is now tied to disease. That information is important, and we can share that back to interested participants. In summary, Genome Connect is working to engage patients in collection and sharing of genetic and health information. Data sharing is important to increasing our understanding of genetics and health, and it also can provide benefits back to patients and registries. With that, I want to acknowledge all the people that helped make Genome Connect and the ClinGen patient data sharing program possible. While I was unable to present in person today, we welcome questions via email at info@genomeconnect.org.
So this was a great overview of what those ClinGen surveys are and how they related to ClinVar. We developed a workflow on the back end for curation of the genetics reports that you share. So Ashley, as a trained clinical genetic counselor and variant curator, she will be able to see if those variants are new or not, and we can then report de-identified information to ClinVar, which may help future patients to be diagnosed more easily and more quickly. In addition to that, what have we learned so far? So we kicked off the SDS-GPS project in the spring with a pilot group of users, so about 15 or so participants, and had them go through all the surveys. And based on their feedback, we made some improvements. Not so much changes to the content of the forms, but just the workflow to make the workflow easier. And then we opened up SDS-GPS to more people, and now we have 30 participants. So I can share with you a little bit of the types of learnings we already were able to achieve with the GPS program. Next slide, please. So we have about 30 participants now. About 20 of them are in the US, and the rest are elsewhere, so Canada, Australia, and Europe. I covered up Europe because each country may just have 1 or 2 participants, and so I covered it up just to protect the participants' privacy. But as we get more and more participants in each country, it will be a really cool visual to see where our participants are throughout the world. Next slide, please. So, for example, this is a result from the Health and Development Survey, so the survey that Alexis just talked about. The question here was, have you been told that there were issues? Have you been told that there were issues during your biological mother's pregnancy with you? And about half said yes, and half said no. Next, please. Do you have issues with your skin? 55% said yes, and the rest said no. So it might be that skin issues are actually quite common in SDS, right? So it might tell us to look further what those issues are. Next, please. Have you had behavioral or psychological issues? 37.9% said yes, 55% said no, and 7% unsure. So that is quite significant, right? Close to, well, let's say about a third of patients do have issues with behavior and psychological issues. So again, this is something that we may want to look into further to see what type of issues are happening, what interventions are helpful, which ones are not. And we may be able to help families this way immediately, and then we can also impact future therapy development opportunities. Next. Have you had issues with your heart or blood vessels? So just wanted to show you an example of something that is not that common. So only less than a third have some issues with heart or blood vessels, and the majority of patients don't. Next. Now, when it comes to blood and bleeding, then that's different, right? Now, more than half of people have issues with the blood. That's not surprising, SDS being a bone marrow failure disorder. Next. If SDS-GPS research team learns about any potential updates to your genetic testing results, would you like us to contact you? 100% of participants said yes. Next. Research interest. Would the patient be interested in providing a blood sample or other biological sample for research purposes in the future? Again, the vast majority of patients are interested in helping out this way, and just a few were not interested in this opportunity, in such opportunities at this time. Next. Would the patient consider participating in clinical trials in the future if asked? 70% said yes, 30% said maybe, and nobody said no. So again, this just indicates that people who joined the GPS program are probably interested in helping improve future therapy opportunities for the community. Next. And then we also had some open-ended questions, and I just wanted to share what that would look like on our end when we analyzed the data. So the question was, what are the most important questions that you have about SDS or related conditions? So people who are interested in learning more about leukemia, liver issues, other medical aspects. And so we see answers in this way and then can analyze them and learn from them and really understand what is important to you all. Next. We can also generate word clouds. So this was a similar question. The question was, if you had a magic wand, what aspect of SDS or related condition would you fix first? And not surprisingly, most people are concerned about the marrow, the risks, the fear, bone marrow failure, cancer. Those types of words and issues came to the top. Next, please. So these slides were really just to show you that in just a short amount of time, with just 30 patients, we already were able to learn so much. And this really solidifies what we should focus on and what aspects of SDS life we should communicate to researchers and regulators and drug developers to make the future better. So I think that's it from my end. Originally, we were planning to show a video about sibling research and open up the conversation to just connect with each other and chat. I will hold the video for another time or share it through the resource section of the meeting platform. But we can just open up the floor now for coffee chat, discussion, connecting with each other if people are available. It has been a long day, so it's up to you. I'm gonna stick around for at least a couple of minutes and go from there. So thank you everybody for coming. We are doing all this for you, and I really hope you got a lot of information and hope out of all this work that the whole community has contributed towards. So thank you, everybody. Thank you, everybody, for coming.